Hello students, welcome to Vedantu NEET English. I am Ms. Gopika, your own biology master teacher. So I hope I am audible, visible and the presentation right behind me is also visible. I will also let everyone know in the telegram that the class has started because of the technical issue. We just got it fixed. Okay. I am just hoping and praying that the screen doesn't trip again so we can have a peaceful class. Yes, 1 second students, just informing them. Chalo, yes. I have already mentioned what was the issue on Telegram. So those students who are unnecessarily, making drama on the chat, I do not understand why you guys are making drama, students. It is technical issue. It can happen anytime. Okay. So I have already informed on Telegram that the screen was tripping and they were trying so hard because my class was the only one that is there. Okay. So yes. So Sukanya, I have told the reason. Okay. Sukanya, Sony, all of you, I have already told the reason. Okay. Yes. So students, listen, biotechnology, we are going to do the lecture 2 part of it. So last class we did up to understanding what are the tools required for recombinant DNA technology. Today we are going to do an overview and also we are going to understand different types of plasmids and how do we use them and etc. like that. Okay. Yes. Okay. Fine. Good. Okay. Good. One second, let me refresh the chat so I can see all of your chats as well. Okay, yes, awesome. Awesome. So students, quickly mark your attendance as well. Okay, sometimes happen, students, big things happen, no, in small places. I mean, sorry, in big places, small things happen. So like that it happened. Okay, but luckily my team was always there here and they helped. Okay, chalo, let's start the class. So last class we did up to plasmid. Okay, last class we did up to plasmid. So this is going to be my new look for everyday's class because of eye issues. Yes. What is it? Ideal Spets 24. Any issues you are facing? Okay, chalo, nice energy. I know all of you were waiting and that's why you got a little angry. Ma'am, where are you? We are waiting for you. I totally understand, students. Even I was waiting. I was feeling bad. Okay, I did not have an answer to tell like what happened. Even I didn't know what was happening. Okay, so yes, now we are back. So we should be happy for that. So students, we'll start from PBR 322 because I have to tell a topic which is connected to this, which I did not tell in the last class. That is insertional inactivation, which I told just like a line. So today I will make you understand this vector once again and then we'll go to insertional inactivation. Okay. So I was trying to make a trick out of this, but the trick was very funny. So I'll tell you the trick. Okay, you people are going to laugh at the trick. So students, to remember what are the selectable markers and what are the cloning sites on each of the selectable markers, I just thought of a random trick. You just tell me how it is. So tetra, sal, bama, amplified, physical science teacher, that is your PT teacher, and PV University. I don't know if you guys can remember this, but I felt like, I feel students will find it very difficult to remember which are the cloning sites on the selectable markers. Yes or no? Okay. Yes. So students, look here. What is going to happen? Okay. What is going to happen is, students, look here. You people tell me how is the trick first. Tetra stands for, okay, tetra stands for tetracycline resistant, sal 1, okay, the cloning vector, bama is BAMH1, okay, amplified is ampicillin resistant, PST teacher is your physical science teacher or your PT teacher, okay, PT teacher, and PV University, everything I only created. Okay, if you can remember this, it is very easy. Why? I'll tell you, because you have to know PBR 322 by heart. Okay, they are going to ask you anywhere, maybe here they'll say, let's say here they put of A, okay, here they put B, they'll tell identify A and B. How will you identify if you do not know the cycle? Okay, so understanding is important, but at the same time you have to learn these things knowingly, and especially they are going to ask you what are on the selectable markers. So selectable marker is ampicillin resistant and tetracycline resistant. So on ampicillin resistant you are going to have PST1 as well as PVU1, okay, and on tetracycline you are going to have SAL1 and BAMH1, okay, BAMH1. Got it? Done? So this you try to remember. If you cannot remember, then students, you will have to learn it by otherwise, okay. So yes, so PBr322, okay, PBr322 is found because of the name, okay, because of the name. So this is Bolivar and Rodriguez. They were the people who found it, okay, Bolivar and Rodriguez, okay. And the number, this was formed in the lab, okay. So this is the number, okay, this is the number. Now this plasmid, see what and all should be there. It should have an ORI site, yes or no? It should have a selectable marker. It should have cloning site. Is PBr322 following all of this? Yes or no? Is it following? It's a plasmid, and is it following all of it? Yes, it is following all of it, because last class we understood that yes ma'am, this is following everything, because why? They have an ORI site. What is ORI site? Origin of replication. That means where the replication should start. Now since I taught you MBI, that is molecular basis of inheritance, and now that I am teaching you biotechnology, you will be able to connect, yes or no? You'll be like, okay, ORI site was there in the replication, okay. Now students, at the same time you have repressor of primer, okay, that you can just leave it here, but understand these are the people who are going to make the proteins for your replication, okay. Now here, these are the selectable marker, that is ampicillin resistant and tetracycline resistant. Now the question is not going to be this. The question can be whatever I asked you, any one of this they can ask you. The question is going to be, if I am using this, okay, if I am using this, imagine I am going to add my gene of interest. What am I going to add? My gene of interest or desired gene, okay, desired gene here in the tetracycline resistant portion. So here I have added my gene of interest, okay, here I have added my gene of interest, and students, this question keeps repeating. So okay, this is my gene of interest. What I have done in blue color is my gene of interest, okay, desired gene. Now what have I done? I have basically damaged the tetracycline resistant, yes or no? That is, students, I have made this region intact. Why? Because I have added my desired gene here. Now such a thing happens. If such a thing happens, what are you supposed to do, or what will be the situation like? How will I understand? So they are telling that if I am using a plasmid or if I am using a selectable marker, I will be able to differentiate between three categories. What are the three categories that I'll be able to differentiate between? I'll be able to differentiate between your transformant, that is the one where the vector has gotten, okay. So let's say this is the one, okay, where the vector has gotten, and this is there. So this is a transformed, okay, this is a transformed and recombinant, okay, recombinant cell, okay. Now this is one situation. This is what all of you are looking for, yes or no? Now all of the students in my class is like, ma'am, we have to make a recombinant DNA. I want to get what I want to desire. This is what my ultimate goal is. So this is what we are expecting. But there can be a lot of cell where the transformation did not happen, right? I can get a cell where there was no transformation at all. That is, the vector did not even enter. So this will be what? A non-transformed cell, correct? There could be a cell, okay, there could be a cell where only the vector got in, okay, only the vector got in without the desired gene. So this will be what? It will be non-transformant. No, this will be transformed but non-recombinant. Is this clear to all of you? Yes or no? Okay, non-recombinant. Non-recombinant. They are telling that I can identify these three types of cells. Okay, I can identify these three types of cells with the help of a plasmid like this who has selectable marker, that is antibiotic resistant gene. With the help of a selectable marker, I will be able to identify three types of cells. Yes, students, energy is very low. I don't know what is wrong. Okay, very disappointed in the crowd as well. One second. Okay, Yes. Thank you so much. Okay, students, so this is what we are going to see. Okay, so they are telling that with the help of an antibiotic, okay, with the help of an antibiotic resistance, okay, resistant, selectable marker, I'll be able to differentiate all these three people, okay, all these three people. So let's understand. Now, when I told you, okay, when I told you that, thank you so much students, when I told you that I added my desired gene into the tetracycline resistant region, what did I do? I did insertional inactivation. Did I make those thing inactivated? Yes. Did I make those thing inactivated? Yes ma'am, you made it inactivated. Now as I told you, there are 3 cells, okay, 3 cells. Now you should tell me, okay, where what will be working. Let's take 3 cells. So let's take cell A, okay, this is cell A, okay, here this is the vector, okay, this is the vector. So what is this students? This is a transformed, that is the vector got transformed, okay, vector got inside, but this is non-recombinant. Why it is non-recombinant? Because the vector went alone. He forgot about the desired gene. He forgot the role of himself, okay. He forgot an event, so let's label this as A, okay. The next one is going to be B. B, this is a cell, okay, this is the vector, okay, this is the vector, and here this is the desired gene. Can you all see the colors? Okay, this you can see the colors. No, the orange is not visible. Wait, I'll use another color, students. Green. No, green will look same. Yes, blue is visible, no? Yes, that came with, okay, that came with the, what is that? The desired gene. So this is going to be, students, this is very, very, very important. Okay, you cannot even think of, skipping this topic. This question will come. So please listen. 5 minutes, don't concentrate anywhere, just try to listen to me. Okay, students, this is going to be transformed and recombinant. Yes ma'am, it is going to be recombinant because it came with the gene of interest. Okay, it came with the gene of interest. Now the last category, that is going to be your C. C is going to be what? This is going to be the cell. Okay, this is going to be there. It did not even transform, students. No transformation. Okay, no transformation. This became non-transformant. Okay. Now I am going to grow all of these 3. Okay, first thing task is going to be, I am going to grow all of these 3 cells, okay, in an ampicillin resistant medium. Okay, what am I going to grow them in? Ampicillin resistant medium. Ampicillin resistance medium. Now you people tell me, which all will get transformed, which all will not get transformed. Okay, let's draw a box. Let's draw a box. Okay, this is the box. Let's, okay, very important, listen carefully. Then later don't tell ma'am question came, I didn't know what to answer. Okay, you listen now, you will be able to answer. Okay, after this I'll give you PYQs, that time you should be in a position to answer. Okay, chalo. Now tell me, cell A. Okay, A is there, B is there, C is there, and here, okay, here ampicillin resistance, okay, here ampicillin resistance, here tetracycline resistance. So tell me, students, A, tell me A, okay, will okay, so listen, A, students, A will grow in an ampicillin resistant medium. Yes or no, I want answers. Okay, yes or no should be the answers quickly. Yes, very good. Ampicillin resistant media, yes, agreed. Okay, next I will grow it in tetracycline resistant media. Okay, but I want answers from you people. What do you think? Will it grow in ampicillin resistance or not? See, if the vector is there, the vector, that is, let's say your PBR322 is there, selectable markers are there, they have ampicillin resistance, then definitely it's not going to be affected, so it is going to grow. So think like that and tell me. Okay, yes or no should be the answer. Quick answer should come. Tetracycline resistant. Okay, tetracycline resistant. Okay, first one is transformed but non-recombinant. First one is what? Transformed but non-recombinant. Okay, it will pakka grow. Yes, it will pakka grow. Why? Because PBr322 is there, selectable markers are there, ampicillin is there, tetracycline is there. So it is going to grow, ma'am. It is going to grow. Is it going to grow in the tetracycline medium? Yes, it is going to grow in the tetracycline medium as well, because the selectable markers are there. Tetracycline resistant gene is there, ampicillin resistant gene is there. So these two portions are there, so it is going to grow. Okay, it is going to grow. Why? Because they did not go with the desired gene. We did not do insertional inactivation here. So it is going to grow in both of the medium. Any doubt in this? If you do not understand this, you are not going to understand rest of it. Okay, so tell me, understood? All of you marked your attendance, yes or no? You have to religiously do it, mark your attendance. Okay, it's a habit that you have to inculcate in yourself. Okay, now chalo, B. It got transformed as well as recombinant. That means it went with the gene that I want. It did not forget the gene and go. It went with my gene. So is it going to grow in ampicillin? Is it going to grow in ampicillin? Okay. Anyone did not understand? Please raise your hands. Okay, raise your hands. Come on, students. You people should be the Josh. You people should be the energy. Talk in the chat box with me. Okay, answer. Answer. Only if you answer, you are going to break this barrier of offline versus online, online versus, you know, all this. You have to break the barrier by talking to me, okay? Now you people know me, you're very close to me, so there shouldn't be a thing that, oh, if I answer wrong, will ma'am scold? I'll scold you, but after that I'll correct you, no? Yes. So next question. I want next question answer. Answer should come. Okay, Sukanya did not understand. Dikshi did not understand. Okay, listen to me carefully. Many of you did not understand. Let's go once again. Okay, I have enough time of my life. I'll make sure that I teach you people and then go. See, students, this is a benefit of having live class also, because later you cannot ask me, right, in replay. So first thing, transformed non-recombinant. Let's understand the term. Transform means what? Vector has got inside. Vector got inside the host, but it does not have the desired gene. That means what? If desired gene is not there, students, then this portion is going to be active, this portion also is going to be active. So if I grow it in ampicillin also, they are going to grow. If I am going it in tetracycline also, they are going to grow, because they are resistant to both of them. So the first case is going to be transformed but non-recombinant. That means the vector got in without the desired gene. Okay, without the desired gene. Got it? Yes. Now, second category is transformed and recombinant. That means what I wanted, what was I doing from the start of the class? I was trying to take the desired gene, add it, and make sure that I'm going to get it, right? So I'm going to add it into the host. Now, students, the vector got into the host with the desired gene. That means what? I added this desired gene near the tetracycline. I added the desired gene near the tetracycline. So here I did insertional inactivation. Okay, understood? So transform recombinant means vector with the desired gene got into the host. Okay, as I wanted. Okay, now non-transformation means what? Vector only did not get in. Vector went somewhere else. Vector did not get into the cell only. Maybe the cell host wasn't, sorry, not the cell host wasn't competent. Could be any reason, but the vector itself did not come in. Now I'll ask you a question. Okay, first question is, first option number A. Transformed non-recombinant, will it grow in ampicillin resistant medium? Yes, it will grow in ampicillin resistant medium, because here ampicillin resistance is there. Okay, will it grow in tetracycline? Yes, ma'am, it is going to grow in tetracycline, because it went as a whole vector, it did not get damaged, no insertional inactivation happened. Are you all understanding? Are you all following me now? Yes or no? Tell me, those who raised your hands, are you following me now? It's not opening. Jaldi. Yeah, vector went somewhere. Okay, vector went out with his girlfriend. And no, vector went in, students. Vector went, but desired gene went out. Okay, desired gene went out. Thank you, Sri. Yes, understood. Okay, chalo. Now, next, if you are able to answer the next one, that means you have thoroughly understood. Okay, next question. transformed recombinant. That means what? Imagine each of you were in the lab and you added your desired gene. You added your desired gene in this tetracycline, in this selectable marker where, right, your BAMH1 SAL1 is there. So here the desired gene was added. Now that means what? Insertional inactivation has happened. That means when tetracycline was there as it is, it was going to grow. But when I added something and I, okay, split it, inactivation happened. Now you tell me, will it grow in amphicoelin medium? I want those students who told I did not understand to answer. Very good, Madhumati. Yes, very good, Madhumati, understood. I want other students to understand and tell me, yes ma'am, it is going to grow in amphicoelin medium because we did not do insertional inactivation in amphicoelin. But what about tetracycline? Yes or no? Will it grow in tetracycline or not? Okay. All of you understood? Yes, perfect, perfect. Ma'am, it will not grow in tetracycline because we did insertional inactivation. Okay, we did insertional inactivation and because of that, it will not grow in tetracycline medium, right? We kind of damaged that whole selectable marker. We cannot tell damage, it is the job, but still, I'm telling you to make you understand, we just altered it. We altered it. So what its job was, that is not going to do anymore. Okay, chalo. Now tell me about this one, students. See, the vector only went off, students. Here, there only desired gene went. Here, vector only went off. Vector is like, hey, I'm not coming for this job. So vector only did not get in, or the host and the vector had a fight and they're not ready to allow. The host is not ready to allow the vector inside. So this is non-transformant. Will it grow in amphicoelin? Will it grow in tetracycline? See, you should remember that PBr322 is not inside this fellow. Okay, yes, perfect. It will not grow in both. So I am going to not even see if it does not grow in amphicoelin itself. I am going to reject this fellow outside. Okay, if here when I am checking, I am noticing that this fellow hasn't grown, I am going to reject him then and there. Okay, I am not going to keep him for tetracycline. Okay, I am not going to keep him for tetracycline. So understood? So only your A is going to grow in both the mediums, okay? B is going to grow only in amphicoelin because their insertional inactivation did not happen. C is not going to grow in both. Perfectly clear? Okay? Yeah, both it won't grow, okay? You people are more sad than this one. So this is called as insertional inactivation. That means on that selectable marker or where the cloning site is there, if we add a desired gene, that means the function of it is going to get changed. Okay, because of which when we do this. Now, why did I teach about this? I taught you that selectable markers are helpful in selecting transformant, non-transformant, recombinant, non-recombinant, yes or no. I also told you that I will teach you the difference between transformant and non-recombinant. Now you would have understood. Transformant means if the vector has got into the host, transformation has happened, because transfer into the host is called as transformation. If that much has happened, we will call it transformant, but recombinant we cannot tell. When we will tell recombinant? When the desired gene also came with the vector. Okay, desired gene also came with the vector. Got it? Chalo. Okay, this is clear. So this question will come, so you should be able to answer. If they give you any cell where they give you TNR, you should understand transformant, non-recombinant. Okay, this question has come in previous years. Okay, so make sure that you understand. Chalo, see here, what is insertional inactivation? See, selection of recombinant due to inactivation of the antibiotics is a cumbersome procedure because it requires simultaneous plating on two plates having different antibiotics, like how I did amphicoelin and tetracycline. Okay, amphicoelin and tetracycline. So therefore alternative selectable markers have been developed which differentiate recombinant from non-recombinant. Understood? Okay, whatever you studied only, we're just reading NCERT because tomorrow when you read, you shouldn't have that doubt. Okay, now on the basis of their ability to produce the color in the presence of a chromogenic substrate. In this, that is, students, we can use a dye. Okay, we can use a dye. Now I'll teach you that when I'm teaching you about your PUC 18, I'll teach you about a dye, how this chromogenic dye, how is it seen and all of that. Okay, but right now you just focus here. Now see, in this a recombinant DNA is inserted within the coding sequence of an enzyme beta galactosidase. This is what I'm going to teach you when I teach you PUC 8. Okay, in PUC 8 what happens? PUC 18, they have not told about it, but here see, they have mentioned about this. Okay, so I'll come back to this paragraph once I teach you that. Clear to all of you? Once I teach you that, I'll come back to this. Okay, chalo, the next plasmid we have to learn. Okay, next plasmid is called as TI plasmid. I think all of you know the full form, no? Yes, jealous. Sukanya, I will go in my order, dear. Please be relaxed. Okay, please be relaxed. I will go in my order. Gel electrophoresis is going to come. I'll teach you in a different order. Okay, I'm not going to teach you in an order that everyone follows. I'm going to teach you in an order which you will understand. Okay, so trust the process. Yes, we tell no, trust the process. Chalo. So students, Agrobacterium tumefacium. Okay, oh, this board has started. Agrobacterium tumefacium. Okay. Now Agrobacterium, this everything is dancing its own way. Okay. Now students, Agrobacterium tumefacium. Now see, Ti plasmid. Okay, Ti plasmid. This, how does this agrobacterium get into the plant? That's a question, no? So yes, Ti plasmid full form is very good, tumor inducing plasmid. Tumor inducing plasmid. Tumor inducing plasmid. Now students, how does this enter into the plant? How many of you in your, okay, yeah, gene tabak tum tum only is happening on the board. Students, how many of you in your small age, you know, have written, you know, like you would have carved on the tree, you know, with the pen and pencil to show someone that you like, you know, S heart, G, Y heart, Z, and all of that you would have written on the plant if you have done it. Okay, most of you don't do it, but then if you would have seen it, no, on the different trees it will be written with blade and all, you know. Shah Rukh Khan loves, you know, Gauri, and Shah Rukh Khan loves Deepika. So like that, it will be written on the trees. Now that is a wound. Okay, that mark that you make. Okay, that mark that you make is a wound on the plant. Okay, that wound, through that wound, students, this Agrobacterium tumefacium, this tumor-inducing plasmid will enter. Not plasmid. This causative agent is going to enter. Okay, this causative agent is going to enter through those wounds. Okay. This is Tare Zameen Par's dance is happening. Letters are going wherever they want. So this wounds is the entry point. Okay, this okay, this wound is an entry point for you know, all this Agrobacterium tumefacium, which will happen mostly in your dicot plant. It happens mostly in your dicot plant. Got it? Dicot plant. Now students, dicot plant, it happens. This causes, okay, this causes something called as your crown gall disease. What disease? Crown gall disease. Crown gall disease. Is this clear? So Agrobacterium tumefacium gets into the plant with the wounds that you make. Okay, because of you people, the wounds that you make, I'm not telling all of you, okay, but you people would have seen, no, some people will be there who shows all their love and they think they're Romeo and Juliet and this tree is going to be everything for them. Okay, this tree is going to walk and go all the way to the girl and show that, see what he has written for you, and vice versa. So that wound, through that wound, this will enter inside, and especially this happens in dicot plant and causes a disease called as crown gall disease. Okay, now ma'am, why are you teaching about disease here? Because students, we are going to use this plasmid. This is called as disarmed. Okay, we are that means we are, you know, removing the problematic fellow, and there we are adding our desire gene. I'll teach you. Okay, I'll teach you this. Okay, now look here. I hope this doesn't dance. Now this is your Ti plasmid. Okay, look here. This is your Ti. Students, I'll just write this downwards, okay, so I can use this place. Okay, just tell me, this one is caused, okay, this plasmid, let it be there. Okay, I'm going to write how this is, you know, brought in because of a wound, okay, especially seen caused in your dicot plant, okay, dicot plant, and causes crown gall disease. Actually, what happens is there, students, this one will get in, it will release your cytokinins and auxins and do continuous cell division and then start forming tumors. Okay, that is exactly how the mechanism happens. So what they get into the wound, okay, then they start releasing cytokines, okay, cytokines plus auxins, okay, and they will continuously do cell division, okay, they'll continuously do cell division. And that's obviously, continuous cell division leads to the formation of tumor. That is how they will form tumor, and that tumor is your crown gall disease. Okay, so in a plant you will be able to notice, okay, this is the plant, you will be able to notice a swelling like this, okay, swelling like this, okay, this is a tumor, okay, this is a tumor. Clear? Clear? Okay. Exactly, that's a belief that if I write your name on this tree, we are going to live forever. Okay, after that 2 days later they'll forget of the name only. So students, look here, this is the whole thing. Okay, this is the, what to tell, the TI plasmid. Now ma'am, why are we talking about TI plasmids, or what is the whole process? See, understand that TI plasmid, okay, TI plasmid have a tRNA in them. Okay, TI plasmid have a tRNA in them. Stay. Okay. Okay, what do they have, students? A tRNA. Okay, let's just do this portion as the tRNA. tDNA, okay. Is it visible to all of you? 1 second, I really can't see because of reflection. Let's change this. Okay. Okay. Yes. Reflection is there, no? Reflection is still there. Can't do anything. Okay. Can't do anything. Can't do anything. Okay. Yes, tDNA, students, not tRNA. Okay, by mistake. Like, teaching molecular basis, I've continuously telling tRNA, tDNA. Okay, tDNA. Yeah, here. Okay, this tDNA will be there. Okay. Now, yes, this tDNA will enter into the plant. Okay, this tDNA will get transferred. Okay, transferred into the plant cell. Transferred into the plant cell and cause the infection. Okay, cause the infection. Got it? Now, ma'am, what are we going to do with that? Okay, what are we? Yes, tDNA only, dear. tDNA only, I just told, okay, by mistake. So, what are we going to do with this, ma'am? It's a problematic fellow. Why are you using this? Very simple, students. I am going to remove this tDNA. Yes or no? I am going to remove this tDNA here. From this plasmid, I am going to remove the tDNA and I will add my desired gene there. Then it is no more problematic. Yes or no? Agree, all of you? If you feel that this is problematic, now say let's one student is there in the chat who is very problematic. If I remove that student, then the problem is sorted, no? Problem is sorted. So here, removing the tDNA, I will add my desired gene. I will add my desired gene. So rest it will do, no? Rest it will do, no? This is all it. Okay, this is how you are going to use this. So I will add my desired gene, okay, after removing the tDNA. Understood? Removing the tDNA. So this is the whole about this TI plasmid. So TI plasmid is called as tumor inducing plasmid. And how do they cause the tumor, students? So this is called as, okay, this is called as tumor inducing plasmid. Okay, tumor inducing plasmid. This tumor inducing plasmid is used, okay, as a plasmid for what? Okay, plasmid for what? That we will be able to use this because it has the ability to take our desired gene. But how? Actually, how do they work? They are agrobacterium tumefacium. I've added the image, don't worry. Through a wound that you made on the tree, it will enter inside a dicot plant and causes a crown gall disease. Now, how will they cause a crown gall disease? It is because of the release of cytokinins and auxins. Okay? Yes, very good. Understood. Cytokinins and auxins, and this cytokinins and auxins will do continuous cell division and form a tumor. Okay, and form a tumor. They will form a tumor, and that tumor is going to cause this ground gall disease, okay, which is like a swelling, on the plant. Now, people understood that, okay, let's do one thing, let's remove that tDNA who is actually getting transferred into the plant cell and causing this infection. Let's remove that problematic tDNA, and there let's replace it with a desired gene. Okay, let's replace it with a desired gene. Yes? Yes, remove that fellow, then he'll come from another account, I know that. Okay, strong. Okay, spammers means you should be strong. You should not be like, oh, one account I have. She blocked me from one account. No, no, you shouldn't be weak, no. You should come with another account. So, is this clear to all of you? If you don't understand the students, again, question always, a lot of questions come from TI plasmid as well. Okay, TI plasmid as well. Now, look at the diagram here, you will understand. You will understand the diagram. Is it visible clearly? Yes, chalo. See, agrobacterium tumefacium. So, this is the TI plasmid. They have a tDNA and restriction sites. Okay, restriction sites. Now, what we are going to do? We are going to insert the desired gene. Okay, we are going to insert the desired gene. So, insertion of gene into the plasmid using restriction enzyme and DNA ligase. Same process. You will cut it with the same ligase, so you get complementary ends, then you add it. Okay, then you add it. After adding it, students, this recombinant TI plasmid will be formed with the desired gene, but not with the tDNA, which is problematic. Then we will introduce into the plant cell in the culture, and regeneration of the plant will happen. So, the tDNA carrying the new gene within the plant chromosome. Understood? So, we will basically inactivate or remove this tDNA and add there a desired gene, and that place this so this will keep continuing without causing any harm to the plant. Okay, so plant we will get with our desired trait. Is this clear? Very simple. You cut something, add something, and then just insert it somewhere. Okay, insert it somewhere. Okay, yes ma'am, clear. So this is the image. Okay, someone is asking for the image. No, this is the image. So look here. Vectors for cloning genes in plants and animals. You may be surprised to know that we have learned the lesson of transferring genes into plants and animals from bacteria and virus without which have known this for ages. How to deliver genes to transform eukaryotic cells and force them to do what the bacteria or the virus want. For example, agrobacterium tumefacium. Okay, a pathogen of several dicot plant is able to deliver a piece of DNA known as tDNA to transform normal plant cell into tumor. So, the problem fellow is who? tDNA. Okay, he is able to do the tumor and direct these tumor cells to produce chemicals required by the pathogen. So, similarly, retrovirus in animals have the ability to transform normal cells into cancerous cells. Can you believe the retrovirus that we kept studying in HIV is also used under this technique? Because students, retrovirus have the ability to do everything. So, what we will do? We will disarm this retrovirus and add our desired gene and tell this retrovirus to do whatever you are able to do. Okay, so we are using their abilities to cause disease, converting it to a good thing. Understood? Converting it to a good thing. So, we are also going to use your retrovirus. So, here see, a better understanding of the art of delivering genes by pathogens in their eukaryotic host has generated knowledge to transform these tools of pathogen into useful vector. So, these pathogens have been used into useful vectors. Retrovirus is able to become cancerous, that means they have the ability to form tumor. Now, what we are going to do? We are going to disarm whichever is the one who is ability. Let's say enzymes are there. No, now HIV I am talking about, but generally other retroviruses you see, they have the ability to convert DNA to from RNA to DNA. So, we will disarm that and we will add our desired gene and we will tell you continue. Okay, you continue whatever you want to do. So, for delivering genes of interest to humans. The tumor-inducing plasmid, that is your TA plasmid of Agrobacterium tumefacium, has now been modified into a cloning vector, which is no more pathogenic to plants but is still able to use the mechanism to deliver genes of interest. Okay, genes of interest. So first you listen to my explanation, then you read NCERT, it will be perfectly clear. Okay, but first you should listen to me. Okay, first you should listen to me. Yes, dicot plants, okay, not monocot, dicot. Aha, all are plants only, no? Then all of you are humans only, no? So like all are living organisms. So we'll, from tomorrow onwards, we'll start calling each other monkey, telling that hey, you are also living, even I am living. Sony is telling what all are plants only, no? Be it dicot, be it monocot. So from tomorrow, everyone can call random names, okay? Because all of us are living. We all have DNA. We all live, breathe, move. Okay, so see, similarly, retrovirus have been disarmed, okay, and are now used to deliver desirable genes into the animal cell. So once a gene or a DNA fragment has been ligated into a suitable vector, it is transferred into the bacterium or the plant host. Okay, we will transfer it. Okay, we will transfer it. That is what we are going to talk about. Okay, now one more plasmid we have to study, students. Okay, one more plasmid. First, I'll draw the plasmid. Till that, you people can not do anything else, just listen. Okay, yes. Okay, so this is your PUC 18. Okay, what is PUC 18? Plasmid. Okay, plasmid. It is found in University of California. Okay, University of California. Okay, and 18 is the attempt. Okay, this is the attempt that they made to find this. Okay, this is the attempt. Got it? So this is the name, university. It is found in University of California, and that was the attempt. Okay, brightness is too much, dear students. Yeah, no, brightness is little too much, I feel. 1 second, let me try to reduce it. Okay, now I think, is it better now? The white is just radiating. Now it's better, no? Yeah, better. Better. Okay, so yes, this is the one. Now, ma'am, why are you teaching us this? You cannot ask me that question first of all. See, they are going to have two regions, okay? This is going to be a ampicillin resistant region. Okay, we are not going to talk about this region, so you do not have to break your head telling that, oh my god, this ma'am again, she's going to teach about ampicillin. Don't take tension, we are not going to talk about this fellow. Okay, ampicillin resistant is here. This side is where the important person lies. Okay, the important person is going to be here. That is none other than your lag Z. Lag Z or lag Z. Okay. Okay, this is the portion where your lag Z is there. Now this also carefully listen, then only you will understand that insertional inactivation paragraph. Otherwise you will see the entire solar system when you are reading it. Okay, chalo. See, this is PUC 18 and this portion has ampicillin resistance. This portion has what, students? Lag Z. Now my students are going to tell me, where did you see lag Z or lag C somewhere? Okay, where have you seen this lag Z? In which chapter have you seen lag Z? Lag word also, where have you seen? Okay, multiple questions I've asked you. You should answer me all of it together now. Tell me lag Z, where have you understood? Who's going to answer first? Okay, Madhumati has told something. I'm expecting something else. Okay, I'm waiting for some other answer. Where did you learn the topic? That's all you tell me. Okay, just tell me, where did you learn it? Yes, very good. Who told first? Sneha, Madhumati, Rohit. Okay. Very nice, very nice. Okay, yes. So, ma'am, we learned this in molecular basis of inheritance under Lac-Operon. Now, students, carefully understand, this LAC-Z is going to release what? Beta-galactosidase. Okay, it is going to give out beta-galactosidase. Beta-galactosidase. Now, this LAC-Z or your PUC18 has around 13 cloning sites. Okay, what did you hear from me? 13 cloning sites. That is, I'm going to write few of the cloning sites. Okay, we don't have to remember all 13 students. Okay, this is just to make you understand. Even though it is not there in NCERT, there is a topic given, but they did not give the explanation for it. Okay, they have so many of them, like your HIND 3, okay, PST1, ECO R1, okay, ECO R1, then they have SAL1, okay, they have PVU1, okay, so many they have, so many, you know, cloning sites they have, so many cloning sites they have. Now, when there are so many cloning sites at one region, we call this as multiple cloning site, okay, we call it M cloning, okay, MCP, okay, we will call it as multiple cloning sites, right? Why did I write P, students? S, multiple cloning site, okay, MSP. This is multiple cloning site. So, generally, if beta-galactosidase is given out by your LAC-Z, I am going to give a substrate to understand if my LAC-Z is inact or did it have an insertional inactivation. That is what, okay, that is, if I am going to add my desired gene here, insertional inactivation will happen, yes or no? Yes, ma'am, insertional inactivation will happen because we learned it in tetracycline. Now, students, to understand if insertional inactivation happened or not, we are going to use some chromogenic dyes. Okay, chromogenic dyes means the dyes which are going to show us the color. Looking at the color, we will be able to understand if this got recombinant or not. Okay, so listen to me carefully. This LAC-Z is going to give out beta-galactosidase. We are going to do a one-step process where I am going to give X-gal. What am I going to give? X-gal. This X-gal is a substrate. This X-gal is a substrate. Now, all of you know substrate enzyme complex, that is enzyme and substrate, if it comes together, it will give a product. Okay, so here this is the enzyme, I am going to get a product. Okay, I am going to get a product. Yes, I am going to get a product. Now, to confirm if I got or if this beta-galactosidase used up the substrate to form a product, I am going to use one dye. Okay, I am going to use one dye. What is the dye I am going to use? I am going to use a chromogenic dye. Okay, that's all you will tell. Okay, a chromogenic dye. So, in this chromogenic dye, if two colors it can give. If the LAC-Z, okay, has beta-galactosidase or the LAC-Z is intact, that means I did not put any desired gene or they did not get anywhere recombinant. Okay, did not get anywhere recombinant. Many, these people are, I mean not these people, all of you have liked, I know, but new students have to join. Okay, all confused, all are sleeping at home thinking that okay, December we will study. Okay, we will use a chromogenic dye, students. Now, what is going to happen in the chromogenic dye? I am going to give you two instances. Okay, two instances. Now, choosing color is the biggest task. Okay, this is a Petri plate. Now, you are going to see some colonies. Okay. See, you can see some white colonies and some places you will be seeing the blue colonies. Blue colonies means you consider this as one. Okay, you consider this as one white spot. Okay, like this many will be there, so they will form a colonies. Okay, now you can see some white, some blue. Some places you will see white colonies, some places you will see blue colonies. Now, ma'am, what is this white and blue? You did not tell us what is this white and blue. Very simple, students, very simple. See, if there is LAC-Z and the LAC-Z is intact, okay, intact. That means they have not got recombinant or no desired gene has done insertional inactivation, then definitely beta galactosidase will be released, definitely XGAL is going to be used, enzyme and substrate is going to form a product. If that happens, it is going to show blue color, okay, it's going to indicate blue color. It is going to indicate blue color when there is non-recombinance, okay, non-recombinance, or when the lag Z is intact, okay, intact. Did you understand this? All of you tell me in the chat, did you understand this, how you are going to see the blue color? Okay. So actually a lot of people are waiting for December, because I had put a poll on my Telegram, Biology by Gopika, all of you can join the Telegram. What happened? They around, I think 16% or 18% of the students have answered, I am going to start my preparation in December. They are guts to start a NEET exam preparation in the month of December with having only January, February, March. I understand you can join crash courses as a revision, but you cannot depend the whole syllabus, right? Whole syllabus, because during December we'll be doing one shots and we are going to be at fast pace, because now we are going very slow, right? We are teaching every single detail and again explaining. For those students who are not understanding, we are ready to explain again, but this we cannot do in December, no? So students, look here. So the ones which are blue in color, the lag Z is going to be intact. That means substrate and product formation has happened. Okay, product formation has happened. Now ma'am, what about the white? What about the white? You guys tell me, what about the white? The white is going to be when an desired gene is added here. Okay, let's say a desired gene has got added here, and that desired gene has make sure that lag Z got you know, lag Z is not going to release the beta galactosidase. Okay, why? Because insertional inactivation happened. Because of that, this formation also will not happen. So that will give us the white color. Okay, that will give us the white color. So white color indicate that recombinant DNA has been, okay, recombinant DNA has been added. Okay, recombinant DNA is been formed in this plasmid PUC18. Clear? Clear? Clear, 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 clear. All of you tell me. Okay, that is lag Z has got recombinant. That is lag Z is no more intact. We have added a desired gene there, and because of which lag Z is going to become recombinant. Okay, so that is going to form the white color. That is going to form the white color. Pakka? All of you? Now we will go. Okay, yes, recombinant happened. Okay, recombinant happened. So if the product formation happens, just remember this, product formation happens is blue color. If product formation is not happening, it is white color. Okay, if product formation happened, it is blue color. If product formation does not happen, it is white color. That's all. Simple. Okay, simple. Okay, is this clear? Okay, I've picked up all accents from you people. This is PUC18. Okay, PUC8 also is there. 8 has around 9 sites. Okay, 8 has 9 sites. Again, base numbers are different. 2, 6, 8, 6 base numbers these people have. That one has different. Okay, so students understood? These are the colonies. Okay, these they are going to form colonies. Okay, I'm not happy with any of the colors today. Today is a very bad technical day, students. Technically, it's not a good day for me. Okay, they are going to form colonies here. So these colonies will help us identify if recombination happened or not. Okay, so blue will show that the lag Z is intact, recombination did not happen. White means recombinant DNA has been formed because the product formation did not happen. If the product is forming, it's going to be blue in color. If the product is not forming, it's going to be white in color. Done, 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 all of you. Yes or no? Okay. So, and students, just don't forget this MCS, that is multiple cloning sites. Can you see multiple? I have only written few of them. There are around 13 sites which you don't even know. Okay, names you don't know. So, we have written only few of them. Okay. Yes, ma'am, understood. Now, students, this one, see, so many sites are there. Okay, so many sites are there. And I think PVU is in there, no? Instead of PVU, we'll write something else. Okay, yes, so you can see here, see this is an MCS site, lag Z site, lag R binding site, that and all you don't have to bother. See, I told you 2, 6, 8, 6 base pairs are there, that is ampicillin. There's an ORI site also. Okay, we'll write an ORI site also. ORI site. Okay, ORI site. Obviously ORI should be there for everyone. Okay, unbelievably people are telling me only to move on my own diagram. I will not move. Just joking, students. Hello, Divya. Why, Mani? Is it not simple? It's not simple, huh? Very simple, no? You just have to remember Laxi, you know beta galactosidase, you know XGAL. XGAL is the only thing that you are giving for it. Okay, it's a one-step process. So you're giving XGAL. If XGAL and the enzyme, that is enzyme plus substrate, forms a product, you're going to get blue color. If product formation does not happen, it's going to be white in color. Blue means intact. Laxi did not get the desired gene, no insertional inactivation happened. Laxi is staying like that. But if white is formed, that insertional inactivation has happened. Okay, insertional inactivation has happened. Now students, look here. Now we'll go back to that slide which I told you. Okay, I told you now, I'll come back to this. Now read this, you will understand. Okay, so this is the problem, students. You need a teacher because see, beta galactosidase is mentioned, but you do not know such a plasmid. You have never even learned about such a plasmid, so you will be confused. Okay, what is this insertional inactivation? What is this chromogenic dye? So that's why you get this. Okay. Okay, so look here, students, a chromogenic substrate. So here what is happening? Therefore, alternative selectable markers have been developed which differentiate recombinant from non-recombinant, that we understood, on the basis of the ability to produce a color in the presence of a chromogenic substrate. Okay, in this a recombinant, so chromogenic dye, instead of that you should use the word chromogenic substrate, because that XGAL, no, that is the reason why this whole color is happening. Okay, so you can use the word chromogenic substrate. Okay, yes, in this a recombinant DNA is inserted within the coding sequence of an enzyme beta galactosidase. This results into inactivation of the gene for synthesis of this enzyme. Understood? Laxi gene will be inactivated, so it will not form the enzyme. If enzyme is not formed, they are not going to form the product. Okay, so which is referred to as insertional inactivation. The presence of the chromogenic substrate gives blue colored colonies if the plasmid in the bacteria does not have an insert. Understood? If I did not insert my desired gene into the Laxi, I am going to get blue color. Okay, students, so that word you change. Okay, don't write chromogenic dye, you write chromogenic substrate itself. So you'll remember it's XGAL. Okay, now presence of an insert results into insertional inactivation of the beta galactosidase gene and the colonies do not produce any color. Clear? Yes or no? Now you understood? Because that time if I would have taught you, you wouldn't have understood. That's why I thought I'll teach you after teaching this. Okay, any doubts? Okay, I'm writing here also substrate, so later it's easy. Done. Okay, chalo. I understand. In school and colleges you cannot expect this kind of explanation, students, that you can only be able to study like this. Okay, in school and colleges they will think about the mass. Okay, they will think about finishing the lectures. That is what they will be thinking about. Okay, now we are going to come to all this gel electrophoresis. Okay, Sukanya will be excited. Now students, I actually took a different technique. You would have understood that I did not go in order. I took a different order by itself. Okay, I took a different order by itself. So what are the steps in recombinant DNA technology? That means what is exactly happening in recombinant DNA technology. But to be frank, you already have learned around 6 steps. Okay, 2 more steps you have to learn. So we will go in order. I will teach you every step again. Okay, which is like a revision. At the same time, I will also add on to some steps like gel electrophoresis, PCR. Okay, some vectorless, insertion. All that I'll teach you. So the steps involved in DNA technology, students. First thing that you have to do is, see, if you have a problem, first you have to identify the problem. No, so like that, identification. Okay, identification and isolation of DNA. Are you all enjoying this chapter? Is the chapter boring? Is the chapter difficult like you thought? Okay, how many of you are studying this chapter first time, like online or whatever? Okay, first time who is studying, or who has studied in school and studying it for the second time. Before that you have never read. Anyone? Anyone? Quickly tell. Okay, By then I'll write the second step. Second step is going to be, students, fragmentation. Okay, fragmentation by who? By your molecular scissors, which is your restriction enzymes. Okay, restriction enzymes. Then what, after fragmentation, what you're going to do? Okay, by then all of you can answer. Yes, thank you, Dikshi. Okay, thank you so much, so sweet you guys are. Okay, so the next thing is going to be, we have to do separation. Okay, so we're going to do separation by which method? By gel electrophoresis. Okay, by gel electrophoresis. All of you are waiting for gel electrophoresis, no? I will teach you. Okay, gel electrophoresis. Then students, after gel electrophoresis, we got a piece of DNA. Okay, we got a piece of DNA. What we are going to do? We are going to multiply the DNA. Okay, we are going to multiply the DNA. That is going to be called as amplification of your amplification by PCR, polymerase chain reaction. Okay, polymerase chain reaction. Students, let me also tell you guys this. I think 2023 one question had come. What they are going to do is, they are going to write all these steps. Okay, around 7 to 8 steps they are going to write, and they will ask you to rearrange. Okay, rearrange. Every day I have to punish one student then. Okay, yes students, so you have to know these steps in order. Okay, think of it like that. First I have a problem, so I'll identify the problem and I'll isolate myself. Then I will do fragmentation. That is, I'm going to think, is it my job, is it my relationship, is it my family, is it my studies? I'm going to do fragmentation. After that I am going to separate my problems and understand by the help of gel electrophoresis. Then I will amplify my problems. Does not make sense. Okay, so I'll stop it here. Okay, when I started it, it was nice to hear, but later it became tragic. Okay, so yes, fifth step is going to be ligation. Ligation is, you have to attach these gene fragments. Okay, ligation. After ligation, what you will do? You have to transfer into the host. What is the use of just sticking and keeping? You have to transfer into the host. So you will do transfer into the host, and that process is called transformation. Okay, transformation. That's how you study transformant and non-recombinant. Okay, recombinant. Okay, after you transfer into the host, what are you expecting for? You are expecting it to be cultured. Okay, so culturing the host. Okay, culturing the host DNA, and we go to the last and the final step. After culturing, students, we are going to get the desired gene. Now I want to make it into a mass scaling, so I'll be doing upstreaming and downstreaming process. So we will do downstreaming process. Okay, downstreaming process. So can you remember the steps, students? Okay, can you remember the steps? Abhiyosh is like 10 minutes I have studied. Now I need 15 minutes break. Yeah, Sony B went for out of station, okay, for pollination to, okay. So much stress is there, students. Stress, okay. If otherwise it will go to inbreeding depression. So it has to go outside, it has to migrate. Yes, listen here. First step is going to be what? Identification. So identification and isolation of DNA. Then we will do fragmentation by the help of restriction enzyme. Then we will do separation by gel electrophoresis. Then we will do amplification. Okay, amplification by polymerase chain reaction. Yes, then we will do ligation of the gene. Then we will transfer it into the host. After that we will culture the host DNA. Then we will be like, oh, by making this 10 cultures I'm not going to be happening. Okay, then after that we will do downstreaming process. Okay, downstreaming process. Yes, Sony B is going to give case on all of us. Okay, telling that ma'am and the students are punishing me for attending class 5 minutes late. Okay, yes, such a regular student, waited for 20 minutes and she was sending continuously messages telling, ma'am, start the class, ma'am, I'm waiting for 2 days. Okay, I'm waiting from Tuesday. So she is my favorite student. Okay, so you guys cannot tell anything. I can tell anything, but you cannot tell anything. Understood? Yes, this is called partiality. Okay, be a teacher and then do this. Okay, steps is clear. Now we'll go one by one. If step is not clear, you should tell now. Okay, one step we have to go. Identification and isolation of DNA. What am I going to do? I have to identify the DNA. That what is the DNA that I want? What is the desired character I want, right? First, I am going to do, take this DNA. Students, which was the example I took? Yeah, let's take Jimmy and Sammy. Okay, Jimmy and Sammy. So with the help of Jimmy and Sammy, what I am going to take? Okay, I need to take their blood sample, and I cannot take the RBC or the platelets, because platelets is actually a mega karyocyte. That's a big cell has got broken down and then it has become, right? But what about your RBC? RBC is not nucleated. If nucleus is not there, where will you find the DNA? Okay, so we'll have to go for, okay, we have to go for your WBC. Okay, now students, we'll take the blood sample of Jimmy because we need to take the gene of interest from him, right? So we will take the blood sample. Okay, we will take the blood sample, and from the blood sample we are going to get into the nucleus. From the nucleus we will extract the DNA, not the full DNA. We will extract what? The desired gene from it, right? We are going to spot it, we are going to isolate it. Okay, we are going to isolate it. Now this I told you, okay ma'am, but what about if it is in bacteria? What about the cell membrane? No, we have to dissolve the cell membrane. So what we have to do? Okay, yeah, Harshain, it's very sad, but it's a good class. Okay, later some students will realize. Okay, Animal Kingdom, Tuesday. Okay, these people talk in Tamil and Telugu thinking that I do not understand. Okay, I just have difficulty in reading. Okay, Sunday I might not take students. Okay, Sunday students, 1 day break. Okay, Sunday don't ask me to take. I'll take on Tuesday. Okay, I'll take on Tuesday. Yes. Did Sony go away because of us? Find, spot Sony. All of you call and she should attend the class. Okay, chalo, look here. Now if it is a bacteria. Okay, now if it is a bacteria, what will I use? I will use lysozyme. Okay, lysozyme. Okay, because I need to enter into the cell membrane. Okay, I have to dissolve the cell membrane and enter inside to get in till my nucleus, right? So same way, if it is a plant cell, then what I'm going to use? I'm going to use cellulase. Okay, if it is fungus, I'm going to use chitinase. Yes or no? These are the things I'm going to use. Okay, these are the things I am going to use. Okay, yes, clear? Okay, so students, look here. Bacteria, we will use lysozyme. For plant, we will use cellulase. For fungus, we will use chitinase. Now, you are telling me, ma'am, I only want the DNA. I do not want RNA. I do not want protein. I will listen to you, students. Okay, because obviously, I will listen to you. So, what I am going to do? I am going to take, okay, this is the cell. Okay, let's say RNA is there. To remove this RNA, I am going to use RNase. Yes or no? I am going to use RNase. We have studied in Avery, McLeod and McCarthy's experiment that we can use enzymes to degrade them. Now imagine there are protein molecules. Okay, there are protein, different polypeptide chains are there. Okay, polypeptide chains are there, and you are telling me, ma'am, I don't want the polypeptide chain. It is disturbing my extraction of DNA. I will still listen to you and I will tell, okay students, I am going to use a protease, right? I will use a protease to remove the protein. Is this clear to all of you? Okay, now who is the one who is going to use a DNase? Okay, who is the one who is going to use the DNase? Whoever uses the DNase will get hittings. Okay, because you want to extract DNA and you add DNase that will degrade the DNA. Then what are you expecting, students? What are you expecting? Okay, so do not add DNase, understood? So you add RNA, you will remove the RNA, right? RNA gets degraded. Okay, you use protease, you are removing the protein. Protein gets degraded. protein gets degraded. Got it? Yes. Okay. Neelam, you cannot see such a big person standing in front of the board and teaching, huh? Yes? You're asking anyone here? Here. Not able to see, I guess. Do you need my glasses? Okay, look here. This is done. Now, from Jimmy, I am only going to take desired gene. Does that mean Jimmy is going to die, students? Tell me. Will I kill Jimmy because I am taking the desired gene? Yes or no answer? Okay. Will Jimmy be alive? This is the desired gene that I want. Okay, this is the desired gene that I want. Desired gene that I want. Hi Tanuja, what happened? Yes, all of you? Clear or not? This is clear? So this is the desired gene. Now tell me, I have taken this desired gene for Sammy. Will Jimmy die because of it? Okay, will Jimmy die because of it? Faster. If you people tell it will die, no? Then that's all for you people. End of the day. No ma'am, it will not die. So students, now I have taken the DNA sample. Okay, now I have taken the DNA sample from Jimmy for Sammy. Okay, so this is the first test tube I am going to use. Okay, this is the test tube. Okay, this is one more test tube. So when I took the DNA fragments from Jimmy, my DNA fragments were like this. Okay, scattered. My DNA fragments were scattered like this. Got it? So this is my DNA fragments. I took the gene, the DNA was looking like this. Now what will I do? To that I am going to add something called as the chilled ethanol. What am I going to add, students? I am going to add chilled ethanol. Okay, chilled ethanol. This chilled ethanol will do the job of condensing this whole DNA into a single, like a mesh. Okay, so it will condense this and it will make it like a solid. Okay, like a, I don't know how to tell the word. Okay, like a mesh. Okay, this process of using this is called as spooling. Not schooling, okay? DNA will not go for like use. It's called spooling. S-P-O-O-L-I-N-G. Okay? Yes. Thread-like suspensions will be seen here. After that, we will make it into a clump. Okay? Like a clump, it will be there. We will add chilled ethanol and it will, it will get condensed and it will cause spooling. Perfect? Yes? Not the drinking ethanol, the other chilled ethanol from the lab. Now, what has happened? First step is over. I identified it. I isolated it. I isolated the DNA. Now, if it is bacteria, I will use lysozyme. If it is plant, cellulase to get into the cell wall. Okay, fungus, I'll use chitinase. If I want to remove RNA, I'll use RNase. If I want to remove protein, I'll remove protease. I'll use protease. If I want to remove DNA, you cannot remove DNA because you have to use DNA. So do not do it. Okay. So then we will do spooling. Okay, blood sample is taken from the nucleus. We will take the DNA's desired gene. Jimmy is not going to die because we took the desired gene, because we are taking a small portion of the gene. Okay, small portion of the gene. Understood? First step clear? First step clear? Okay, 2 minutes, 1 second water break. All of you drink water as well. Students, we have a celebrity. Do you want me to welcome?
Glasses?
Yeah.
Fancy.
Not fancy. I can't see anything, by the way.
It looks fancy though.
Yeah. I bought it on special request from my students. Because the other one was not matching enough. No, not exactly. This they gave me.
Scam.
Students, welcome the celebrity with lot of fire emojis. Okay?
Well,
The students are so mean they are becoming day by day. They're telling me 1 second is over, ma'am. I have been waiting for 24 minutes, ma'am. This got tripped. Both the studios, okay? So continuously around like 4 or 5 times. Yeah, full thing. the OBS went, everything went, and the students are like, ma'am, you should at least tell me the reason. It's like, you know, we tell the principal, no, that, sir, I want to take a holiday because I am not feeling well. So I should have told them, it seems. And I told them in Telegram, and they did not see the message, and they're scolding me. They're like, ma'am, at least you should tell the reason. We are waiting for 2 days. From Tuesday, they are waiting, it seems here.
Oh,
Papa, they've become old waiting for 2 days.
Yeah.
Yes, Diksha ma'am. Sneha is seeing you as Diksha ma'am. I cannot believe.
So the thing is, once what happened, I'll tell you the story behind it. Okay, Diksha sir. So what happened was, one of the thumbnails, they put my image in my session itself as Diksha ma'am in the bottom. So my image is there, it's written Diksha ma'am in the bottom.
That's why they keep telling Diksha ma'am when you are here.
Exactly. From that day onwards, it has started a trend. Because I started laughing because even I didn't know, I'm like, Diksha sir, what? And from that day onwards, everyone has caught on of it. Like,
that's sir, nice, yeah. Should I start implementing? And then let's call Diksha as Diksha. No. You're Diksha sir. So she'll be Baswa ma'am.
No. We'll make you Shreyas sir. We'll make you Shreyas sir.
No, we'll call her Baswa Sauruses ma'am. Okay, students?
It has changed so much. They're calling me Dosa sir also now. It has changed suddenly everything flipped one day.
Now tomorrow, like, you know, stressors will be there, I'll be here, and then we'll be doing work, energy, power, or current, electricity.
Exactly.
Then they'll
chemistry, we'll be teaching physics.
Yeah, then they'll start calling Capto ma'am. And they'll call sir attendance sir.
Attendance sir.
Conjuice sir. Randomly imagine sir start getting message, Conjuice sir.
Next time when Shreyas sir is taking class, right? Conjuice sir. Sir will like, what conjuice?
So I'm like, what did I do?
Exactly.
What happened?
Exactly.
Did I do something wrong? So
yeah. I don't know.
He keeps telling all this nonsense, students. Do not listen. Please stay motivated. Be in one track, okay? You saw, no? All this crown gall disease, all of that. Okay?
The plant, no?
Don't talk.
Yeah, so I was calling them also plants now. Because plants are smart sometimes, so I'm trying to call them smart, motivating.
Oh, that's why they started waking up in the morning to do photosynthesis. They're like, now they don't keep alarm anymore. They tell mom, when sunlight comes, I will wake up by myself.
Remove the curtain, I wake up and I eat first.
I'll do photosynthesis. Oh, okay? Chalo.
So when we finish,
yeah.
I'll have to take one more class now.
Okay, so bye.
Bye-bye.
Yes. So, students, Diksha sir has left. So, Baswa ma'am, we will welcome next class, okay? Yes. All drama you don't do. You're learning biotechnology with zoology, ma'am, okay? Don't tell I am plant, I am that, this and all, okay? You're all animals now for me. Can I tell that? Okay? Yes. Okay, now you learn zoology or else now it's there for you. Okay, now I'll make learn well, exactly. Okay, so otherwise your parents will call you animals. So isolation of genetic materials, students, understood? So isolation of genetic materials, recall that nucleic acid is the genetic material of all organism without exception. This is the introduction I gave you for molecular basis of inheritance. Now, in majority of organism, this is deoxynucleic acid or your DNA. Yes, 90%, 99% of the organisms, the genetic material is going to be, you know, deoxy, that is DNA. Okay, only in viruses you will see RNA, but RNA is very useful because it's going to act like a catalyst, messenger, adapter. Okay, adapter. In order to cut the DNA with the restriction enzyme, it needs to be in the pure form. Okay, so free from other macromolecules. Since the DNA is enclosed within a membrane, we have to break the cell open. That's why I told you, if it's a bacteria, we will use lysozyme. If it is, you know, fungi, we will use chitinase. If it is a plant, we will use cellulase. Okay. Okay. Yeah, today actually less animals have come, students. Usually more animals will come. Okay, I was taking class on CBSE and the amount of animals that I met there is like very nice. Okay, they're like just coming and going, and I was like, okay, I felt like a zookeeper for a minute that time then. So what to do? Now, since the DNA is enclosed within the membranes, we have to break the cell open to release the DNA along with other macromolecules such as RNA, proteins, polysaccharides, and also lipids. So for all of this, we can use lipases, we can use, right? RNAs, we can use protease, we can use all of this, we can use. Okay, understood? Now, this can be achieved by treating the bacterial cell, plant cell, or the animal cell with enzymes like lysozyme, I already told, cellulase, chitinase. Okay, you know that genes are located in a long molecule of DNA and we'll have to take a small portion. You will not take a 2.2 meter long DNA. Okay, yes. Done, 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 done. Next topic. Yes, next topic is what? Fragmentation. Okay, now we have selected the gene. Okay, we have isolated it, and this is the gene from Jimmy. Okay, Jimmy the cow. Okay, you have to make Jimmy famous, students. So learn properly and answer all the questions, then only Jimmy will become famous. Okay, now this is the gene. Yes or no? This is the gene. Okay, now this desired gene, I have to make sure that I am fragmenting or chopping it off. For that, who am I going to use? I am going to use a molecular scissor. Okay, a molecular scissor. Who is that molecular scissor? Because I am cutting from the inner part of the DNA strands, I am going to use a restriction endonuclease. Yes, I am going to use a restriction. This is a revision for you as well. Okay, restriction endonuclease. This restriction endonuclease is going to cut my desired gene. Okay, desired gene. Now, one thing that you have to remember very, very importantly is that okay, so students, one thing that you have to importantly remember is, now this desired gene is there. Now my vector is there because I have to add this desired gene into the vector. So let's say this is the vector DNA. I have to make sure that I am cutting both of the vector as well as my desired DNA. Okay, oh my god, what am I telling? Desired DNA. Okay. Okay, desired DNA. Okay, and your vector DNA. Students, I have to cut both of them with the same ligase, right? I have to cut them both with the same ligase. For what? Okay, for what? That is because if I don't cut them with the same ligase, I am not going to get complementary ends. Okay, I am not going to get complementary ends. For me to get complementary ends, we need to make sure that we cut them with the help of the same ligase. This we have discussed when we spoke about blunt ends and sticky ends, right? Whatever you are cutting them, you have to cut the same way. I also showed you paper example, right? If I tear it together, it is going to, all of them are going to tear the same way. But if I am going to tear it differently, I am not going to get the same halves. So same like that, you have to cut them with the same DNA, I mean restriction endonuclease, and then you'll have to, you know, ligate them. Okay, you have to, like, you know, glue them. No, for gluing them also we need to make sure that we get complementary ends. So remember this was a PYQ. This has come in one of the years, so do not forget. Okay, so we will use the same restriction endonuclease, same restriction endonuclease to cut both the DNAs. Okay, both the DNA, and even ligation will be with the same DNA ligase. Clear? Yes, ma'am. Yes, complementary ends. Okay, this is your fragmentation. We have cut them, students. After cutting them, okay, you already know about the palindromic sequence and all of that. No, Malayalam, mom, dad, however you read it, it's going to be the same. Okay, so restriction endonuclease, desired gene you're going to get, they are going to cut at the ends, and we'll make sure that the cutting is going to happen in the same way, so we get complementary ends, so that sticking is going to be easy or gluing is going to be easy. Okay, done. Chalo. Now, cutting of the DNA. See here. So this way what happens is, you will study the recombinant DNA technology, at the same time you will revise what you studied in the last chapter. So last chapter I taught you about the tools, today we are learning about the recombinant DNA technology. Okay, that is why I kept gel electrophoresis to the last. Okay. You people roast, when your marks come I will roast. Okay, cutting of the DNA at a specific location. Look here, restriction enzymes digestions are performed by incubation purified DNA molecules with restriction enzyme. We have to make sure that we get the pure form of the DNA. You cannot have other macromolecules with the DNA. That's why we used all of the, you know, enzymes, okay, with the help of ACE before. Okay, at the optimal condition for a specific enzymes. Agarose gel electrophoresis is employed to check the progression of the restriction enzyme digestion. DNA is negatively charged, so here they are talking about gel electrophoresis a little bit, but still see, DNA is negatively charged. Anyone has doubt in this, okay, molecule, hence it moves towards the positive electrode, that is your anode. The process is repeated with the vector DNA also. The joining of DNA involves several processes. Okay, after having cut the sources of the DNA as well as the vector DNA with a specific restriction enzyme. The cut out gene of interest from the source DNA and the cut vector within the space are mixed and ligated. Okay, ligation is going to happen. So what they're going to say, so here I got the desired gene, right? I got the piece of my desired gene. Yes, see, I got this piece of desired gene. Now I have to cut the vector as well. Okay, I have to cut the vector as well. Okay, nice. You people got saved. Okay, you people, your tongue is there. That's why students, you half of you are surviving. Okay, you're very, very smart. You know exactly what to tell your teacher. Okay, so students, from the vector also I'm going to cut. Now what am I going to do? Let's say this is the vector. Okay, vector. I've cut the vector DNA. After adding to the vector, or let's do one thing. Let's just draw this only. Okay. Okay, now I am going to add this desired DNA, that is I am going to ligate. Okay, this is too hard to draw actually. Okay, I am going to ligate. Students, I have added my desired DNA here. Okay, I have just made a connection here actually, just to show. Okay, just to show. Okay, desired DNA is here. So desired gene was cut. Okay, so what did I cut? I cut the gene of interest, or GOI. Okay, gene of interest. Now I inserted it here. Okay, I inserted it into the vector. Done all of you? Done all of you? Chalo. Okay, fine. Now separation, gel electrophoresis. Now listen to me carefully, all of you waiting for gel electrophoresis. No? Now students, gel electrophoresis principle is very simple. Who is larger? Now I am giving you 10 kg back. Let's take Sneha and let's take Tanuja. Two people are there. Tanuja I have given 2 kg back and I have given Sneha 10 kg back. Now will Sneha run faster or Tanuja will run faster? All of you in the chat box, you can put T or S. Okay, will Tanuja run faster or Sneha will run faster? I have given her 10 kg of atta and I have given Tanuja only 2 kg of atta. Okay, if they are lazy, both of them are not going to run. That is a different story, but other ways. Tell me generally who is going to run. Okay. Okay, who is going to run? Obviously, Tanuja is going to run because she has only 2 kgs of atta, so she will be able to walk faster, she will be moving faster. So, your entire gel electrophoresis work on this principle that the smaller fragments will move farther, okay, and the larger fragments will be at the nearest. Okay, now since DNA is negatively charged, okay, since DNA is negatively charged, what is going to happen? From the cathode, it is going to run to the anode. Obviously, I am negative, I will run towards positive. No, opposite poles attract. So, negative to positive it is going to run. So, it is going to run, and what is going to happen? Smaller fragments are going to run faster. Okay, it is going to run faster and reach that. Larger fragments are going to run slow and it is going to be in the nearest position. Okay, now this happens in an agarose gel. Okay, this is in an agarose gel which is extracted from your seaweed or red algae, we can tell. Okay, now look at the setup. Okay, look at the setup. This is your agarose gel. General principle you understood, no? Now I'll teach you the how does it look and all of that. Okay, since we usually do gel electrophoresis, okay, in our masters and degree and all. So one second. Okay, look at this. This is your agarose gel. Okay, what is this? This is going to be an agarose gel. Agarose gel. From what is it extracted, students? A gel, like a gel. Okay, gel substance you can imagine. What is it extracted from? It is extracted from seaweed. Okay, seaweed. You would have heard this in a lot in Korea, right? When we talk about sushi and all of that, we learn seaweed, no? So this is your red algae. Okay, red algae. It is extracted usually from the red algae. Now, this agarose gel, in the agarose gel, we are going to add our DNA. Okay, we are going to add our DNA. For that, there are going to be wells here. Okay, what are they going to be there? This wells, this is called as the comb. Okay, this is called as the comb. like this. Students, how does the normal comb look? This is how your comb looks, no? If you're using a bigger teeth comb. Okay, this is how your comb looks. So this is the name of it, comb. Now why is this comb there? Comb is there so that you can add your fillings there. So in this here, let's say 1, 2, 3, 4. Okay, 4 wells are there. So these are the wells. Okay, what are these? Wells. In this wells, you are going to add your DNA. Now, since you cannot see your DNA, you can use a loading dye which is blue in color. Okay, so with the help of a loading dye, I am going to load the DNA. Okay, I am going to load the DNA here. So I have loaded my DNA here. Okay, students, otherwise you cannot see if you have loaded it or not, because everything is going to be transparent or, you know, colorless. So you will use a loading dye which is blue in color. Okay, it's actually going to be dark blue in color actually. It's somewhat like this color. Okay, this color it is going to be. So what did you use? A loading dye. So with the help of the loading dye, you have taken or added your DNA. Okay, you have added your DNA with the help of the loading dye and you're able to see it. Okay. Okay. Now students, what is going to happen? What did I tell? I told you the larger fragments are going to stay wherever they are and the smaller fragments are going to move, right? Smaller fragments are going to move. Okay, so it's not E, comb. So here, let's say this is the larger fragment, okay, and this is the smaller fragments. Okay, here also there are some smaller fragments. Here some smaller fragments are there. Here some larger fragments are there. Okay, some of them are there like this. Now imagine the DNA I added, if it is remaining like that itself, that means it shows that it is undigested. Okay, this is one. Okay, smaller, smaller, will keep moving. Okay, like this. Okay, just this is just an diagram of it. So students, look here, look here, this larger fragment. Okay, this is the larger one, this is the larger one, and it has it moved a lot? It has not moved a lot. Okay, so largest it's going to be nearer. Okay, nearer, and the smaller fragments, okay, the smaller fragments are going to be at the far. Okay, smaller are going to be far. Now, so this gel electrophoresis totally works, okay, totally works on the principle of sizes. Okay, who is fast, who is small, so like that it totally depends on the sizes. So the principle of this gel electrophoresis, okay, principle of your gel electrophoresis is based on size. Okay, based on size of DNA fragments, based on size of DNA fragments. Clear? DNA fragments. Now let's say this is how it is going to look like. Now students, I told you it is going to move from whom to whom? It is going to move from your, okay, here is the cathode. So it's going to move from your cathode towards whom? Towards your anode. Okay, towards the anode. Because DNA is negatively charged, so definitely it's going to move towards the positive charge. So it's going to move from the cathode to the anode. Okay, thank you so much students. Yes, so here this is going to happen. So gel electrophoresis, general idea you understood, no? Now I'll show you something. Okay. Okay, look here students, how the loading will happen. See, like this loading will happen. Okay, these are the wells. Can you see? These are the wells into which loading is going to happen. Got it? So to each wells loading will happen. See, if it is not there, okay, once the electricity is going to happen. So see, you can see the movement. Can you see? The smaller ones are running faster and it is moving towards the anode. Okay, so sample 1, sample 2, so we'll keep doing different ones. Now listen, try to understand. Gel electrophoresis means what? We are using a gel and we are using some electricity, and because of that electricity we are getting a separation. That is the meaning of gel electrophoresis. So same thing is going to happen. We are going to add the sample in each of the thing. If we cannot see it, so we'll add loading dye. After that we'll be able to see their movement once the electricity is on. So what they are going to do? They are going to run as fast as possible towards the anode. Smaller fragments will reach faster, larger fragments will be nearer. Okay, yes. Yes, so now look at how it's moving students. Can you see how the moving is happening? That is the loading dye. Okay, that blue color thing is the loading dye. Because of that only we'll be able to visualize it, otherwise you'll not be able to visualize it. Later for visualization we'll use ethidium bromide, that is a different story. But here can you see to understand if it is moving or not, we have to do this process. Okay, do this process. So look here, we'll add the sample. Okay, DNA is loaded. Can you see? Can you see how it's moving? See how it's moving? Okay, so longer fragments, shorter fragments. Okay, these are the longer or the thicker ones. Okay, the larger ones. Yes, okay, perfectly clear to all of you. This is what your gel electrophoresis is. Okay, gel electrophoresis is. So here, so this word, okay, separation by gel electrophoresis, we did the separation, larger fragments, smaller fragments, all of this we did. And also, okay, we also have to understand, it is gel is used, okay, that is your agarose gel. Okay, agarose gel is used, and we are doing electrophoresis. That means we are supplying electricity. Okay, electrophoresis, that is we are supplying electricity. And phoresis means where the separation is going to happen. Okay, where the separation is going to happen. So how many steps have we finished, students, in recombinant DNA technology? Okay, how many steps have we finished in recombinant DNA technology? First we understood isolation, identification of the DNA, then we did fragmentation, then now what are we doing? We are doing separation. Okay, now some more things are there. Now I understood, okay ma'am, this ran and everything, I understood that, but I cannot visualize it, right? I need to make sure that I am going to get my gene of interest. Now how do I get my gene of interest? Very simple, students. Now this is your agarose gel. Okay, this is your agarose gel. Okay, this is the agarose gel. Now different segments I got. Now 1, 2, 3, 4. You added 4 samples into your well. Now how do you know which is your gene of interest? Let's say this one is there. Okay, this one is there. This one is there. This one. Okay, different things are there. Okay. Okay. Now when you're looking at this, you should understand, if you want to identify which is your gene of interest among this, I am going to tell that this is my gene of interest. Okay, I am going to tell this is my gene of interest. Why? Because there is something called as a DNA ladder. Okay, what is going to be there, students? A DNA ladder is going to be there. In that ladder, you will be able to see the base pairs. Okay, you will be able to see that, okay, 1200 base pairs, okay, 2200 base pairs, where it will land. Okay, where it will land. So I have chosen a base pair which is 1200 base pairs. So when I look at the readings, I will be able to understand, okay, my base pair has landed here. That means mine. Okay, that is my that is my gene of interest is having 1200 base pair. So this is how I will be selecting it with the help of DNA ladder. Is this clear to all of you? Yes or no? If you do not understand this, you will not be able to understand how you are going to extract your gene of interest. So this is how we will get our gene of interest. Okay, gene of interest depending on the number of base pairs, because we will know the number of base pairs. Okay, we will know the number of base pairs. Done? See, you don't, Rathas, you do not have to like know the depth, because again wells can be different, because the comb size is going to be different. Okay, so you do not have to know the depth of it, because understand, loading sample will be added, it totally depends, right? We will add the comb, so you can check the depth. Okay, the how much you know, the depth is, it's totally up to you. Okay, totally up to you, you can check about it. I do not have a lot of information about how much each comb, or depending on the comb how much the depth of the wells is. Okay, because it, again, it is depends when they are doing the experiment, depends on them. Okay, that the comb that they use and the wells. Okay, so usually for loading the wells are used, otherwise we will not be able to identify. Okay, otherwise we will not be able to identify. Now students, you understood this. Now the next step is going to be, ma'am, I saw the fragments. Okay, I saw the fragments, that loading sample has happened, but I will have to visualize which is my gene of interest. What is going to happen? Yes or no, all of you? Yes, Abhi, you can check after class. Okay, because last class we did, and this class we just did a revision of it. Okay, this class we just did a revision of it, so you can just go and check that, so that way you will be able to understand. Okay, you will be able to understand. Clear? Don't miss it, okay? Tanya, we are not doing the topic, dear. Okay, to visualize this DNA fragments, okay, to visualize this DNA fragments, what we are going to do? To visualize this DNA fragments, I am going to use a, oh my god, okay, okay, I'm going to use a carcinogenic, okay, I'm going to use a carcinogenic dye. That means if you touch this dye, or if it is coming in exposure to your hand, like to your skin, then it can cause you cancer. That's why it's called carcinogenic dye. This carcinogenic dye is none other than your ethidium bromide. Okay, and you know this question is one of NTA's favorite questions as well, even board question. So remember this ethidium bromide, which you can write it as ETBr. Okay, ETBr. This ETBr is a bright orange color, students. It's a bright orange color. Bright orange color. And this bright orange color, you cannot see, even though I'm telling it is bright orange color, you cannot see it with naked eye. So what you have to do, you have to give UV rays for it. Okay, you have to give UV rays for it. If you give UV rays only, you will be able to visualize your different types of fragments. Okay, so once you give UV rays, you will be able to see bright orange color, and then seeing which is your gene of interest. Now you already identified your gene of interest looking at the DNA ladder. Now you know exactly at which position it is there, right? Now you will do something called as elution. Okay, you will do something called as elution, which is your removal of your gene of interest. Okay, why are we doing separation? To understand where my gene of interest is. So we will do elution, that is basically removal of your gene of interest. Removal of GOI. Removal of GOI. So what how you are going to remove? There is something called as F end of tubes. Okay, a structure like this. Okay, a structure like this. Into this you will remove. So how you are going to do? You are going to do a suction, like a suction. Okay, so you will have a tool like this, and with that you are going to take this gene out. Okay, you're going to suck that gene out. Okay, suction is going to happen. You will take that and you will add it into this. Okay, you will take that and you will add it into this. Is this clear? Okay, one suction you will do, and then you will add it into this. Like this, you will remove it and add the gene of interest here. Okay, gene of interest into this F end of tubes. All of you clear? Yes, no? Any doubts? Okay, so we are separating. Basically, we are separating the fragments and we are just taking it. Saying that, okay, this is time now, let's remove it, and after removing, we will continue. Okay, after removing, we will continue. All of you clear? Pakka clear? Yes, ma'am, pakka clear. Okay, yes, okay, perfect. Okay, chalo, let's continue. Okay, this is done. So now you can add chilled ethanol to this also for doing that spooling, but since I've already taught you, you do not have to worry about it, but otherwise you can remember, okay, where you can add chilled ethanol to this. Okay, you can add chilled ethanol to this. Okay, chilled ethanol, and you can just chilled ethanol, and you can condense this. You can condense this, or like you do that, and then over. Okay, then you will take your gene of interest. Now see, this is spooling, students. Can you see? This is spooling. Okay, you can use a glass. Okay, we usually tell we can use different techniques. You can use a glass tube and remove the DNA fragments. Okay, you can remove the DNA fragments. You can see how they are going to get condensed. So now we have added chilled ethanol. So before they were in fragments. Now after that, what has happened? Condensation is going to happen. So this is called as spooling. Okay, this is called as spooling. So that you will remove it with the help of a glass tube. Okay, to the end of the glass tube, you can add a loop like thing. Otherwise, you can just add the glass tube. So this is spooling. Clear, no? So into the students usually in tubes or even in F end of tubes. Now look at gel electrophoresis diagram. After that, I'll give you a break. Okay. Oh my god, 2:15. Why is time going so fast? When I looked the last time, it was 1:30 or something. Okay, students, look here. Gel electrophoresis, this is your entire plate. So we keep the top plate like this and the current is passed. Okay, now they will start moving towards the anode. Now look at this. This is how it is going to look. Here we will have the we tell a small glass plate kind of a structure. Okay, we'll add that. Into this we'll have some liquids. Here electrodes will be connected, and this is your gel electrophoresis. So here you can see the sides, the end, everything is fully sealed. Okay, fully glass sealed. Okay, fully glass sealed. Now after that, on the top you are going to add the wells. Can you see the wells here? 1 2 3 4 5 6 7. So this is the comb. Okay, this is the comb. Yes or no? The comb is added, then you can add the DNA, and then you can see the larger fragments are on the top, the smaller fragments are moving down. Clear, all of you? Yes. Okay, ma'am. Okay, clear. Okay, now this is your effluent of tube. So what are we going to do? We are going to take this with the help of suction, add it into this. 10 minutes again, transfer of all that spinning and all that is not required. Students, instead of spooling, you can do one more thing. Okay, this is one technique where you condense the DNA and you get it. Otherwise, you can do something called a centrifugation. Yes or no? Centrifugation you can do. That is, you add this. Okay, you add this. The DNA, let's say the whole mixture is there. Okay, the whole mixture is there. What you're going to do? You're going to spin it at some RPM. Okay, spin it at a certain RPM. Set the temperature, everything, spin it at a certain RPM. When you spin it at a certain RPM, what you're going to get? Obviously, you're going to get the pellet as well as the supernatant. No? Like that, you're going to get a condensed form. So here, you will get a proper condensed DNA here. Okay, clear or not? This is clear? Okay, you will get the condensed DNA. So this you can do as centrifugation. So it's not necessary that you always add chilled ethanol. Sometimes we also do centrifugation when there is like a lot of people. We usually in labs and all, like when we are doing the experiment, no, we usually do centrifugation where everyone takes it, adds it, set the RPM. Okay, yes. Draw what, dear? I have drawn everything. Everything is drawn. Okay. So here this is going to happen. So here DNA fragment, centrifugation, it will happen, then pellet will come, DNA will get settled, then whatever is rest will be on the top. Got it? So all the time we do not have to do spooling, students. Sometimes we can do centrifugation as well, and some people do also. Okay, it depends on the situation. Now see, this is the whole technique. What is going to happen? You're loading the sample. Okay, then you're applying the current, then the gel, analyzing the gel. That means it's going to happen. Then loading the DNA. See, this is in your protein. Okay, loading the protein sample. Here you load the DNA sample, you apply the current, and you will see the analyze the gel. Same thing will happen. Okay, analyze the gel. Okay, yeah, you expect more diagrams from me, you expect me to write the entire NCRT, and you people won't attend the class, nor comment, nor like, nothing you will do. Okay, this is one-sided. Okay, one-sided will not go for longer, students. Take these words from me. Okay, take these words from me. After you complete NEET and all, that time if you have one-sided, you will understand how difficult it is. Okay, this is one-sided. I am only doing everything, you are not doing anything. Got it? Some of you are doing, not everyone. Okay, chalo. So I don't know if you people want iPhone or not, but at least write the exam. Okay, don't tell ma'am distraction, ma'am I don't want iPhone. I saw one comment telling ma'am PS5 so much distraction. It's not like as soon as you will write you will get PS5. No, you have to write, you should get the scholarship, and you should get it with so many students. Okay, offline we have lot of batches, right? Offline we have lot of batches in Vedantu right now for NEET and JEE. So all those batch students are going to compete with you. So you have to show our power by writing the exam and getting the scholarship. Okay, student is telling ma'am PS5 such a distraction, iPhone such a distraction. Hello, sabr karo. It's not going to come. All of it is not going to come. You have to get the scholarship. Okay. So write MVSAT, students. Do not take it for granted. October the exam is going to start. So write MVSAT exam and then come back to me and tell that ma'am I got scholarship among all those students. Okay. We have to show the power of our DNA RNA batch. Okay. Yes. Done? Okay. So students, we will resume after a cutie break. Not cutie. Little big. Okay, resume back. After this we have PCR. Don't go back anywhere. Okay, PCR is very important. Resume back Time is going like rocket. Okay, 40, 20 minutes break. Okay, lunch break. Okay, resume back at 2:40. Yes, it's already posted here. All the details about MVSATs, personally, all the details will come on Telegram. Okay, everything about the date, and about the syllabus and where you can write it, offline, online, how you will write it, everything. Okay, yes, exactly. You should show your RNA. Okay, Kuti break is not enough. How long you want the break? Okay, how long you want the break? Enough. This much is enough. See, after that we have, students, PCR we have. After that, you people have to answer my questions. Then we have ligation, transfer. Okay, transfer. Yes, then we have, that's all. Then bioreactor, that's all. Very simple. Okay, very simple. Easily I'll teach you people. Okay, all you have to do is stay here. Don't go anywhere else. Okay, see you all. I'll open the MVSAT link and show you, so you'll be able to understand the details there. All of you come back, okay? Don't go off. Don't run away. Students coming in 2 minutes, okay? Coming in 2 minutes. Just give me 5 minutes, I'll just fill the water in. Okay? Hi guys, I'm back. Yes guys, please give feedback about the YouTube ad, okay? It's very important. Then only we can improve the ads, okay? Chalo, let's start. Yes, welcome back all of you. Did you all have lunch? Okay, ma'am will have lunch after class because there's no time to have lunch. Guys, come on, let's make it 100 likes. All of you, we have to do this, okay? 1 second, call all of your friends. Ask all of them to join, okay? Let me put on WhatsApp as well. I have to tell them break is over, join please, okay? Okay. Okay, so students, please take MVSAT seriously, okay, and do the exam so that scholarship can be offered. Now, so we have done how many steps now? If you go to the steps, we have to see if we have done all the steps. No, we have to do one by one. Oh, here, yes. So students, identification done, okay, fragmentation done, separation by gel electrophoresis. Any doubt in gel electrophoresis, you do not have to study so much in depth, okay? Do not listen to no dear, I'll have lunch after the class, okay, because 20 minutes I cannot buy the lunch. I have to go down and buy the lunch. So students, gel electrophoresis, don't try to learn a lot of things. You do not need a lot of things. Remember about ethidium bromide, the chromogenic, carcinogenic dye. Remember about UV rays. Think about elution as well as the principle of it, that the larger molecules will stay nearer and the smaller molecules will move further, and it moves from the cathode to the anode. Okay, this much only you have to remember. Don't remember too many things also. And all of you, quickly mark the attendance, because I am sure if you have attended this class till now, everything is pakka clear. Yes or no? How many of you agree? Okay, now we are going to do amplification. Okay, amplification means what, students? From 1 I make 2, from 2 I make 3, from 3 I make millions. So I have to make million copies of DNA, otherwise there's no use of doing this whole process, right? At the ultimate, I have to make sure that these people are going to code for the proteins which I need, and I'm going to get the desired character. So just doing one or just doing one set is not enough. I have to do amplification. For amplification, we use a technique called as polymerase chain reaction. What do we use? Polymerase chain reaction. PCR full form. Okay, PCR full form, polymerase. Okay, polymerase chain reaction. Polymerase chain reaction. So this polymerase chain reaction happens in your thermocycler. Okay, that is the instrument used where it is going to amplify your amplify the DNA fragments that I am going to give. Now, how does polymerase chain reaction happens, ma'am? What are the steps involved? Polymerase chain reaction happens in 3 steps. Okay, 3 steps. And you have to remember the order of these 3 steps, because many questions have come every single year about the order of these 3 steps, because students usually just write whatever they want to write. Okay, so first step is going to be denaturation. Okay, denaturation, where you are going to separate your DNA fragments. Okay, second step is going to be annealing. Okay, annealing, where we will add our oligonucleotides. Third step is going to be extension, where we are going to form our new fragments. Okay, now students, you can remember it as day. Day, we tell, no, a day, come here, like in Tamil and all, I think we tell, no, day. Do you use that word? Okay, I've heard, like casually in movies and all they tell, no, day. So like babe, okay, like that, day. Okay, DAE, that is denaturation, annealing and extension. You have to remember it in this order itself. Okay, you cannot remember it however you want, because first, if you do not break the DNA fragments, how are you going to add oligonucleotide? Same way when I taught you replication, I told you our DNA replicates in a semi-conservative manner. First, you had to unwind with the help of helicase, then only you were able to add your DNA polymerase. Same thing goes here. Okay, same thing goes here. So students, see, this thermocycler, or when we do polymerase chain reaction, it's not like at every step you're peeping and looking, oh, denaturation happened, oh, DNA fragments got separated, oh, I'll add the oligonucleotide. It's not cooking, okay? You're not, when you're making tea, you add first milk, then you add slowly one by one thing. The same thing goes with coffee. You add milk, you pour you pour water, then you add coffee powder, then you add sugar, then you mix it. So it is one by one step. That is not polymerase chain reaction. You just initiate, okay? What do you do when you're making tea? What do you add? You add milk, you add elachi, you add tea powder, you add sugar, everything you add, and you boil it at once. Okay, that is how PCR works. That means you just regulate the temperature. Okay, it is boiling, the milk is coming up, you reduce it. Again, when you feel like, okay, the tea leaves are not, you know, I don't know what you tell for that. Okay, basically the color has to change. No, that time again you increase, decrease, increase, increase. Same thing we are going to do here. Here we are going to regulate the temperature, not going to open it and check every single time. Okay, so first step is going to be denaturation. Now denaturation, students, let me see if I have left. Yeah, okay. So these things, now see, temperatures are not mentioned in your NCERT, but usually we all teach the temperature because it is convenient for you to remember that, okay, at these temperatures only changes are going to happen, and this is the regulation that we do. Okay, so first denaturation happens at 90 to 94 degrees Celsius. That is extreme high temperature. Students, can I use DNA polymerase? Can I use DNA polymerase to make my new template? Because I have to make new template, I have to do amplification. Can I use DNA polymerase? Yes or no answer? Can I use DNA polymerase at 90 to 94 degrees Celsius? Yes, no. Fast answers should come, okay? Even if students are less, answers should come fast. Okay, because later a lot of students are going to watch it in replay. So you do your job of answering faster. Okay? Hey, cockroach and frog, I finished? No. How much times I'll do cockroach and frog? I think these people are not asking that. I'll use DNA polymerase. DNA polymerase. Students, khichdi, it will become DNA polymerase. Okay, so much high temperature DNA polymerase can't work. So I am going to use someone who has the habit of staying in hot spring. That is your Taq polymerase. Okay, this fellow is not going to stay in hot spring. From where we extracted, no? Taq polymerase, we are going to extract it from a bacteria called as Thermus aquatis. Okay, Thermus aquaticus. Okay, this is a bacteria that will withstand, or that is, you know, the habitat is, okay, this person's habitat is in hot springs. Students, all these notes you will get it on Vedantu NEET English Telegram as well as Biology by Gopika. So you do not have to worry. All you have to do is understand. Okay, these are present at hot springs. Since they are present at hot springs, will they become khichdi at that temperature? They will not become khichdi. Okay, yes. Tanuja is making something else, students. Tanuja is making for zookeeper, she is adding cockroach, frog and making tea and giving. Please don't drink tea from Tanuja's hand, okay? Save yourself and save this family of yours. Okay, so yes. So look here, Taq polymerase is the polymerase that I am going to use to form my new strands, because DNA polymerase or any other polymerase will not be able to withstand this high temperature during denaturation or for everything, okay? Denaturation is basically splitting your DNA strands, but after that I have to form the new strand. For that they have to survive, because I'll be adding everything at first and then I'll be closing it and then changing the temperature. So Taq polymerase will be the one who is going to be extracted from Thermus aquatis, which is present in hot springs. Done? Okay, next temperature, annealing. Annealing, students, I saw so many different temperatures. Okay, it usually, so I read one of the National Institute paper, they told me that it can go from 55 degree to up to 72 degree, but I won't write 72 degree because extension happens at 72 degree. So 55 to 65 degree is your book value. 55 degrees Celsius to 65 degrees Celsius. Then the last step, extension happens at exactly 72 degrees Celsius. Some people write 72 to 74, but 72 is the actual, okay, actual syllabus. Hi Soumya, okay, I remember you. Okay, yes, students, remember this? Yeah. Dikshi doesn't have family to take it, so Dikshi will drink that, okay, but I will not drink. Denaturation, annealing and extension, 90 to 94 degree, 55 to 65 degree, 72 degree. So what we are going to do, we are going to take the thermocycler, which is the instrument. In that we are going to add the append of tubes. In that we are going to do polymerase chain reaction. We are going to form new fragments. We are going to form million copies of DNA, okay, not just one or two, because in replication and all we are forming very less number. Here we are going to do millions of copies, okay. millions of copies of DNA. Shall we start the process? Okay, start the process. Okay, chalo. First step is what? Okay, yes, first step is denaturation. Okay, denaturation. She's traumatized listening to your tea recipe, Tanuja. Okay, denaturation, what did I tell you? 2 DNA fragments. Okay, 2 DNA fragments. DNA fragment 1. Will I be able to fit all of it here? Okay, DNA fragment 1, DNA fragment 2. Okay, students, they will be together, no? Obviously, they are going to be together. Okay, they are going to be together, and these people are going to have your adenine, guanine. Okay, all of this is going to be there. Okay, all of this is going to be there, and they are going to have bonds. Okay, this might look like a ladder, but it's not a ladder. Okay, they are going to have bonds. Yes. Okay, so like this, bonds are going to be there. Okay, this is how your DNA is going to be. Now what we are going to do? Okay, what we are going to do? We are going to break this, right? We are going to break this bond and separate this bond with the help of what temperature? 90 to 94 degree temperature. The DNA fragments are going to start separating. So denaturation means what, students? Separating the DNA fragments. If you don't separate, how will you make the new DNA fragments? So separation of DNA fragments. Done? She's selling her tea at this point, students. Okay, so 5 dash to 3 dash, we forgot to put this one, and 3 dash to 5 dash. First process is clear? Yes or no? Tell fast. This is very important. Okay, later you will sleep and then you will wake up and be like, oh, you ma'am, when I closed my eyes, you finished teaching so much. Okay, so in this step, students, what is going to happen? Hydrogen bonds are going to break. Okay, hydrogen bonds, okay, will break, and the strands will start separating. Okay, the strands will separate. Okay, done. Strands will separate. Now the second step is called as annealing. Okay, second step is called as annealing. Now students, annealing, the both fragments got separated. Yes or no? That we have already done, that the fragments have got separated. Here one fragment, another fragment. Now to this fragment, I am going to add forward primer and reverse primer. Okay, forward primer and reverse primer, that is to get the free OH group. So I will be adding oligonucleotide. Okay, I will add, so first we will write 5 dash to 3 dash, 3 dash to 5 dash. Okay, what I will be adding? I will be adding oligonucleotide. Okay, I will be adding oligonucleotide. Sorry, here. Okay, you can also call it as reverse and forward primer. If you cannot remember it, it's okay. Okay, if you cannot remember it, it's okay, but at least remember for this one, oligonucleotide. Okay, why? Because we need a 3 OH group to start the joining. Okay, to start the joining. Okay, look here. I think I wanted to show you guys something, but I forgot it. Since 1 second, as a 1 minute, I'll just show you this. This I wanted to show you. I remembered after going there. So this gel electrophoresis, okay, this you already saw. Now, see polymerase denaturation. Can you understand, students? See how unzipping happened. Okay, how unzipping happened. Then new strands started forming. Okay, new strands started forming. Clear? Extension happened. So denaturation happened. okay, at 90 to 94 degrees Celsius. Then annealing is happening around 54 degrees Celsius. What is going to happen? We are going to add oligonucleotides here in the primers, that is at 55 to 65 temperature. Then extension is going to happen where we form the new strand. Okay, look at this. After this, I'll come back to the PCR and I'll teach you. Should I can't slow down, but you see aram se, okay? You see slowly. Fine, okay? Fine. Extension and new strands will form. Like this, million copies will happen, okay? This is only one strand. Like this, million copies will keep happening. Okay, annealing. See, annealing, we added one, this one. Both the sides we added forward and reverse primer. Okay, same thing here, students. See how DNTPs are added. Those are DNTPs, which I'll teach you now. But can you understand? Taq polymerase is making the new strands and forming. Okay, Taq polymerase are forming the new strands. Okay, here you can see temperature is decreasing and increasing. So here, see, we added the oligonucleotides. The green and the brown is the oligonucleotides. This is the forward one. This is the reverse one. So what they're going to do? They're going to keep forming the DNTPs. Okay, they're going to form the DNTPs. I hope no one has any doubts. Okay, then I'll show you an image and I'll teach you the rest. Chalo, okay, understood. Okay, understood. We will go back now. Okay, yeah. So students, see here, this is going to be 5 dash to 3 dash. So this is 3 dash. Towards the 3 dash, I want to have a 3 OH group. Okay, a free OH group here for my continuous bond formation. Now this is going to be what? 3 dash to 5 dash. So this side I am going to have the OH group. Understood? So forward primer, reverse primer. Okay, forward primer, reverse primer is where the OH, where we are adding. So in annealing process at a temperature of 55 to 65 degree, you added a oligonucleotide. So here you are going to write the steps. Okay, you added a oligonucleotide. So you add oligonucleotide. Students, this free OH group I have taught you in molecular basis of inheritance. When I drew the bonds, I told you specifically that free OH group should be there for the continuation, right? That is what we are talking here. So every chapter is connected. You have to keep revising. Okay, add oligonucleotide to, okay, oligonucleotide primer we tell. Okay, oligonucleotide primer to the strands. Okay, to the strands for free 3 OH group. Okay, for the free OH group. Now students, here just I am writing forward and reverse primer. Okay, because somewhere if you see, it shouldn't be in a position like, oh ma'am didn't tell. Okay, forward and reverse primer. Done. This is clear, no? With the GIF you were able to understand, I believe. Now the last step is going to be extension. Okay, last step is going to be extension. So extension means what? Formation of your new strands. Okay, 72 degrees Celsius. What is going to happen, students? To this DNA fragments, we have added oligonucleotides. I think I prefer to draw it thicker. Okay, we have added oligonucleotides. To this oligonucleotides, we will add new DNTPs. Okay, new DNTPs will be added, that your deoxynucleotide triphosphates will be added so that we can form the complete chain. For that, who is going to help? Polymerase we need, no? So polymerase is going to do the job. Okay, so again we will do the DNA strands. Okay, we will draw the DNA. Is it too congested? No, it's fine, no, in one sheet only, because I don't want to take one more sheet for this. Okay, these are the 2 strands, students, and these are my oligonucleotides. Okay, oligonucleotides. Okay, and then you just label it 5 dash to 3 dash. Okay, 3 dash to 5 dash, and then you label this, that is here this is going to be 3 dash to 5 dash, and this is going to be 5 dash to 3 dash. Now what is going to happen? With the help of Taq polymerase and with the help of your DNTPs, okay, with the help of your DNTPs, let's take another color. Okay, these are your DNTPs. Okay, like this DNTPs will be there. With the help of your DNTPs, we will form the new set of strands. So for that, here, right here, your Taq polymerase is going to come. Okay, Taq polymerase is going to come. Here also your Taq polymerase is going to come, and they are going to add the DNTPs and continue this. Okay, continue this chain. Understood, students? Continue this chain. Same thing. So now we got 2 strands. Yes or no? Now we got 2 strands. Yes. Oh, Sony is back. All of you celebrate. Later replay students will be thinking, what is this ma'am telling? Who is this? Okay. So it's a big story, students. Some fun needs to be there in class. Okay. Otherwise, why are we taking live classes? We can take recorded, no? We should have fun in class. Yes. So students, here Taq polymerase. This is Taq polymerase. Okay. This is Taq polymerase. Okay, new DNTPs will be added and this chain will keep continuing. Okay, like that we got a new strand, completely new strand we got. So opposite polarity. Okay, opposite polarity. Understood? Clear? All of you? Yes or no? Easy, no? Very easy, no? I think you would have thought, my God, PCR is so difficult. But is it actually difficult? It is not actually difficult. Agreed or disagreed? Is it actually difficult? Okay. So here hydrogen bonds are broken. Here adding of oligosides, polysaccharide. Here what is going to happen, students? Here new strands are going to be formed, right? New strands will be formed. Like this, we will make millions of copies. Okay, not just one. This is only one we are showing to teach you, but millions of copies we will form. Perfect. Nah, no one is my favorite students. All of you are my favorite students, okay? All of you are my favorite. Whoever attends my class regularly, comments, I know your names, then you are definitely my favorite, okay? I remember my last year batch students also, even though it is online. Okay, I have contact with the students now also. They keep updating me, you know, one student has joined. You know, yes. 3:30. 3:30. Who is there? Oh. I booked na till 3. He'll also take till 3:30. Okay. So students, here what is going to happen? DNTPs are going to be there. Okay, DNTPs are going to be there, and these DNTPs are going to be added to form the new chain. Okay, new chain. Is this clear? All of you? Okay, new strands are going to be formed, million copies are going to be there. Done. Your polymerase chain reaction is done. But if you forget these steps, okay, if you forget these steps, students, then I'm sorry to say it's going to be very difficult for you to remember. Okay, oh no. Okay, very difficult for you to remember. Okay, done. Clear? So look here. So students, this is what the whole process is. Look here carefully. You had DNA strand. By denaturation, we separated the strands. Okay. After that, we did annealing, where we added a forward primer and reverse primer. Okay, forward primer and reverse primer. Did I draw the same arrow mark? 1 second, students. Yes, towards this side, towards this side. Yes, perfect. So forward primer, reverse primer. After that, we start adding with the help of the DNTPs will be added with the help of Taq polymerase, and DNTPs will continue. Okay, and the end point analysis we will do. We will run a gel electrophoresis to see what we have got. Okay, what we have done, is it correct or wrong? Okay, to understand that, again, you will do a you can do a gel electrophoresis to identify that. So this is going to happen in million copies, okay, million copies. And students, to calculate that, okay, to calculate that, you have to do 2 to the power raised n, okay, 2 to the power n. Why you have to do 2 to the power n? Because n, okay, this is how you will calculate. So n is equal to number of copies, n is equal to number of copies, or sorry, number of cycles, so sorry, number of cycles. So imagine if I give you a problem which has 5 cycles, okay, DNA has gone through 5 cycles of polymerase chain reaction, then what is your answer going to be? 2 to the power 5, right, 2 to the power 5. So you will have to do 2 into 2 into 2 into 2 into 2. How much you will get answer? That is going to be the number of copies that we are going to get, okay. So number of copies will be determined by 2 to the power n. N is going to be the number of cycles that has gone through. So like that we will form millions of copies. Understood? Yes, millions of copies. 4, 6, okay, 2 2s are 4, 4 2s are 8, 8 2s are 16, 16 2s are 32. So like this you will keep forming the chains, okay. So this is the your NCERT diagram, students. Here 30 cycles they have given, so that means you will again get it in a bigger number. So 2 to the power 30, okay, you have to do 2 to the power 30, that will give you 30 cycles answer, okay, 30 cycles answer. So this question can come where they will give you the number of cycles and you have to use the formula, okay, you have to use the formula, okay. Maths has come there, all hitting their head. Okay, look here. See, see the enzyme DNA polymerase. The enzyme extends the primers using the nucleotides provided in the reaction and the genomic DNA as template. If the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately billion times, that is 1 billion copies are made. Such repeated amplification is achieved by the use of thermostable DNA, that is polymerase, isolated from the bacterium Thermus aquaticus, which remain active during high temperatures and include denaturation, okay, steps. So this is the, okay, this is the whole process. Students, I have taught you chemistry also. You remember? I have taught you chemistry also. Chemistry I have taught you, physics I have taught you, okay, and now maths also I have taught you so many times. Okay, now ligation of gene with vector, students. Ligation means what? Joining them, okay, joining them. So it is going to happen with the help of DNA ligase, okay, it's going to happen with the help of DNA ligase. Okay, DNA ligase. So DNA ligase is going to do the joining of them. Who? Stitching. Stitching is going to happen, right? So as I told you, cutting and even the gluing should happen with the same DNA ligase, okay. Gluing should happen with the same ligase. Same ligase, because again when we study sticky ends and blunt ends also we learned it, okay. Sticky ends and blunt end also we learned it. Any doubts so far? Yes, any doubts so far? Yeah, ligation is done, okay. Ligation is nothing, students, just ligase, that is gluing, okay. Gluing, you will use DNA ligase, and this DNA ligase is going to glue the ends, okay, glue the broken pieces with the help of ligase. That's all, okay. Ligation will happen. Again, you use the same ligase, like how I told you, same ligase. If you use same restriction endonuclease, you will get same sticky ends, like that only, same blunt ends, okay, same blunt ends. Now most important topic, okay, most important topic. Just give me 1 second, okay. I think one more teacher wants the studio, so I'll just ask him, okay. Yes, students. Today is a problematic day, okay? We cannot do anything. Okay, yes. So, transfer into the host. Now, what is, ma'am, transfer into the host? Students, I told you that we saw no transformant can happen, non-transformant can happen. That is, I am like, okay, vector is set, desired gene is done, I have done ligation of it. Now, I have to transfer this into the host. But host and DNA has a problem. Okay, now why do I say host and DNA has a problem? Let's take this. Okay, this is the cell, okay, bacterial cell. Okay, now this bacteria has a hydrophobic, okay, hydrophobic, so big it is. So this is the bacterial cell. Let's say they are hydrophobic nature. Okay, they are hydrophobic in nature, and then we have our desired DNA, right? So vector is there, okay, our desired gene is there, okay, desired gene is there. Now what is going to happen? Okay, what is going to happen? This DNA is hydrophilic. Okay, what is this? DNA molecule is hydrophilic. Now will hydrophilic and hydrophobic work? One loves water, one is scared of water. Will a person jump into a sea? Okay, imagine Divya is scared of water and Madhya, if I am pronouncing your name right, loves water. Does that mean Madhya will push Divya into the water? No, right? They both will have issues telling that I don't want to play in water, you play in water. So to solve these issues, I am going to come. Okay, to solve these issues, I am going to come. Yes or no? Hydrophilic, hydrophobic, okay, usually no one teaches this, but yes, I have. Yes, okay, bacterial cell is hydrophobic, bacteria, okay, yes, hydrophobic only, bacterial cell, this is bacterial cell, so this is hydrophobic, okay, scared of water. So this bacterial cell will not allow this DNA to enter inside, and this bacterial cell is my host. Okay, host is not allowing. After all the hard work that I've done, host is not allowing, and this DNA is hydrophilic. So what they'll have? They'll have issues between them. To solve these issues, we have to bring in some divalent ions. Okay, we will take the help of some divalent ions, students. The divalent ions, okay, to take the help, we will take some divalent ions. These ions are going to be calcium chloride. Okay, calcium chloride. This calcium chloride is going to help us to get this DNA inside the bacterial cell. Why? I'll tell you. Because students, this calcium chloride okay, this calcium chloride is going to form pores. Okay, going to form pores in this bacterial cell, and because of this pores, it will be easy for my DNA to enter inside. Okay, it will be easy for my DNA to enter inside. Understood? So we will use a divalent ion called as calcium ions to make this host competent. I have used this word before. Yes or no? I have used this word before. Host has to be competent. If host is not competent, right, if you are not competing with each other, then who will compete with whom? Your parents will compete with each other, telling that I gave birth to a NEET aspirant, even I gave birth to a NEET aspirant, so let's fight. No, you have to compete among each other, that okay, I have to study today with Gopika Ma'am, I have to watch the replay, I have to revise, I have to go back and do PYQs. So that competent nature we have to bring in the host, not that this competition, students, but competent nature means allowing the vector to enter inside them, or allowing for the transformation to happen. Okay, so transfer into the host, this process is called transformation. Okay, this process is called transformation. Got it? So we will form the pores so that this fellow can enter inside. Okay, so this will cause competent host. Okay, competent host. Done? All of you understood? Yes or no? So now as the number of pores increases, okay, as the number of pores increases, did you guys imagine your parents fighting, telling that I gave birth to a NEET aspirant? So number of pores increases, it will be easy for the DNA to enter. Okay, to enter. DNA mean I am talking about the vector. Okay, vector to enter. Yes, calcium chloride will act like a mediator. Okay, So process of transfer of exogenously naked DNA into the host, okay, into the host. But students, multiple methods are there, okay. How can we just trust one method? Okay, this is one method, definitely divalent ions is one method where we use calcium ions, but we need to use more methods, right? We have to use more methods. So some of the methods are going to be 4 methods you have to learn, okay, 4 methods you have to learn. First method is heat shock, okay, heat shock. Okay, second method, this ballistic method, or we can call gene gun. Third method is micro injections, okay, micro injections. Fourth method is going to be disarmed pathogens, disarmed pathogens. Ready? Let's go one by one, all of you. Okay, yes, gene gun, yeah, ballistics you can also call. I spoke while drinking. Chalo, let's start with first one. Okay, first one, I think I'll have to duplicate this. First one is going to be heat shock. Sometimes your parents will think that they have to give you heat shock because nothing else is giving you shock, means heat shock only we have to give. What is heat shock? Okay, what is heat shock? You take the bacterial cell, you take everything, DNA, first you add it into ice, okay, then you give heat, okay, up to 42 degrees Celsius, okay, then again you put it into ice. This process will keep continuing as a loop. So bacteria will get shocked. No, oh, sometimes I'm cold, sometimes I'm warm, sometimes I'm cold, sometimes I'm warm. So heat shock will that's not how shock will come. Okay, students. Basically, ice, you dip it in ice, then again you take it and heat it. Okay, in boiling water bath. Okay, we'll keep it in boiling water bath. So how will you give the heat shock? With the help of boiling water bath. So in this boiling water bath, you're going to add the desired DNA and you'll add the bacterial cell, and when the heat comes, automatically the membrane, right, will open and then this will enter inside. Okay? Okay, this will enter inside. I wrote ballistic only. Who wrote biolistic? Okay, I wrote ballistic only. Chitra, you have a problem, I think, that everywhere you will add one extra spelling. Okay? Yes? Didn't I write I? Oh, sorry. I thought she's telling the wrong spelling. Biolistic. Okay, yes, done. Okay, now students, look here. Boiling, okay, boiling water bath. Yes, you corrected, dear. Yes. Okay, so students, what is going to happen? Ice to heat, heat to ice to heat, heat to ice. This will keep continuing and automatically the DNA will enter into the bacterial cell. Okay, this DNA will enter into the bacterial cell. Understood? DNA entered the bacterial cell. Now, the next thing is going to be your gene gun. Oh, gene gun. Okay, or your biolistic. Okay. So here what is going to happen, students? You can use tungsten, okay, tungsten or some gold, okay, and you will pass it on and it will go into the DNA and later the transformation will happen. Now see, we do not give a lot of explanation for this actually, okay, we do not give a lot of explanation for this actually, we just tell, okay, where the like microparticles are there, those microparticles will be allowed to pass on, okay. So if you look at this, okay, this is how your, okay, this is how it will be. So this will be your gold or tungsten, okay, microparticles, we call this as microparticles, okay, microparticles. These microparticles will be allowed, okay, to enter inside. So here what is going to happen, this microparticles will stay here only, here the DNA will be there, The board is also dancing. I am also dancing. Okay, yes, gold or tungsten. Okay, this will be gold or tungsten. Okay, gold or tungsten. The particles will be there, then these particles will be bombarded into the cell. This you don't have to know a lot, but just understand. Okay, microparticles are there, gold and tungsten, and they will be allowed to pass through the cells, and then it will go through the DNA, then to the cells. Okay, so during this time, what happened? The DNA molecule will automatically enter, the desired gene will enter into the cell. Okay, this is the DNA. The DNA, okay, will be bombarded with this tungsten microparticles. Then this DNA will automatically, at that bombarding rate, it will move into the cells and automatically enter into the cells. Okay, enter into the cells. Now students, next one, look at how many lines are there here. Next one is micro injection. Micro injection is nothing, students, directly you inject it into the nucleus. Okay, micro injection is nothing but you directly inject it into the nucleus. So this is the nucleus of the cell, you directly inject it. Okay, you directly do the injection to the nucleus. Understood? This is the micro injection. You just have to know one word of it. Okay, the lot and all they have not explained, only heat shock little more they have explained. Okay, board is dancing. Exactly, it's irritating me actually. I'm getting irritated. Okay, so students, here cells are bombarded with high velocity. I hope all of you know a little chemistry, no? Okay, this Rutherford's experiment, Thomson's model, all this you would have studied, no? So in that and all you would have studied about this microparticles getting bombarded at high velocity, automatically the molecules will pass in. So heat shock done, gene gun done, micro injection done. Next one is disarmed host, okay, disarmed pathogen, that is your both TI plasmid as well as your retrovirus. Okay, disarmed host. Students, why are we doing all this? To make the host competent. If the host is not getting competent, if you are not able to transfer the DNA into it, then your recombinant DNA will not happen. Okay, recombinant DNA will not happen. Okay, you people know to draw injection, huh? No. So here, students, disarmed host is going to be two people, TI plasmid, okay, TI plasmid as well as your retrovirus, okay, as well as your retrovirus. Okay, TI plasmid, what did we do? We removed the tDNA who was causing the tumor. Okay, we remove or we add the desired DNA there. Okay, remove the tDNA and make it do what we want to do. Here also same thing, retrovirus, which was cancerous. Okay, cancerous cells, they can form cancerous cells. We form that also, converted to our own requirement. So what we do, we basically decrease or we completely stop their pathogenic effect. Okay, instead of that we utilize them for our things of adding the desired gene. Is this clear to all of you? Competent, okay, competent host. Okay, the host has to become competent for transformation to happen. Okay, for transformation to happen. Done. Transformation to happen. Yes. Okay. Chalo. Competent host. What is this competent host, ma'am? We have done. So since DNA is hydrophilic molecule, it cannot pass through the cell membranes. Why? In order to force the bacteria to take up the plasmid, the bacterial cells must first be made up competent, okay, to take up the DNA. This is done by treating them with a specific concentration of divalent cation such as calcium, which increases the efficiency with which the DNA will enter inside. Okay, now the bacterium through the pores, okay, it makes pores in the cell wall. Recombinant DNA can then be forced into such cells by incubating the cells with recombinant DNA on ice and then placing them briefly at 42 degrees Celsius. This enables the bacteria to take up the recombinant, because pores are there, it will start becoming bigger and we will be able to enter them. This is not only the way to introduce the DNA into the host in methods known as microinjection. So students, imagine vectorless methods means what? We do not use a vector here, we directly inject the desired DNA into this. Okay, these are vectorless methods. Remember this, that all this microinjection injection, gene gun, all of this, what are we doing? Here we are doing vector-less method, okay? All these are vector-less method. That is, I do not want to use a vector, okay, be it a plasmid, anything, I do not want to use a vector. Without a vector, I will do micro injection where I directly add my desired DNA into the nucleus, or I will do gene gun, okay. So these are the different methods that I take in, okay. So this we will do it in animals, okay. Yes, we will not use the vector. Is this clear, all of you? Yes, and we do this in animal cells, okay. We do this in animal cells, plant cells also some we do. So animal cells and plants. Clear or not? This vector-less I forgot to tell, okay. Something I did. Yes. Okay, now see here, micro injection where the recombinant DNA is directly injected, okay, injected into the nucleus of an animal cell. In other methods suitable for plants, cells are bombarded with high velocity microparticles of gold and tungsten coated with DNA in a method known as biolistics or gene gun, okay. And the last method used are disarmed pathogen vector, which when allowed to infect the cell transfer the recombinant DNA into the host, okay. That is your retrovirus as well as your TI plasmid. Any doubts so far? Any doubts? Now students, we are going to two important techniques called as downstreaming and upstreaming. Now downstreaming and upstreaming means I have done all of this, I want it in a larger scale. So there I am going to use your bioreactors, okay. So I can do a single step bioreactor or I can do a continuous, okay, step of bioreactors, okay. Single step, what happens? One, I'll add everything, I'll add the bacteria, I'll add everything, and mixing will happen, stirring will happen. They will reach a lag phase, okay, they'll reach a lag phase, I'll show you that. Then the other one where there is continuous feeding of the medium, there they will not go through a lag phase, it will always be an exponential phase. This is for like purpose, like I will sell, okay, I will sell something, I will make something, because all this is going to labs, all this is going for experiment. So for that, we have to make sure that we do the downstreaming process where we will check the quality of the content, we will do a proper analysis, okay. Upstreaming means what? Upscaling the quantity. I want to sell it in large scale, I cannot do it in small incubators, I cannot do it in small test tubes and make my multiple copies. So instead of that, I will use bioreactors, okay. So see, when you insert a alien piece, okay, when you insert a alien piece into a cloning vector and transfer it into a bacteria, plant or animal cell, the alien DNA gets multiplied. This is normal. Now, in almost all recombinant DNA technologies, the ultimate aim is to produce a desirable protein. Yes or no? Gene has to become your central dogma, okay. Now, hence, there is a need for recombinant DNA to be expressed. Now, the foreign DNA gets expressed under appropriate conditions. The expression of foreign genes in the host cells understand many technical details. Now, look here. After having cloned the gene of interest and having optimized the conditions to induce the expression of a target protein, one has to consider about making it large scale. That is, once we felt like, okay, let's say Sony and Ratis is going to do this job of selling primers, selling, plasmids, all of this happens, okay. You can buy the plasmids and all of that, as well as you can buy the primers. They have understood that this is the technique that I am using and I am going to get something favorable out of it. If they know that there is something that is going to come favorable out of it, they have to make it large scale. Can they make it in small test tubes selling, hey Ratis, you do 2 test tubes, I'll do 2 test tubes, tomorrow we'll make Kanuja do 2 test tubes. We cannot do that, no, calling everyone and telling, hey, did you do 2 test tubes? We cannot do that. We need big, large scale. We have to upscale the whole process. After upscaling the whole process, we will send it for downstreaming processing where they will be checking of it. Okay, where they will check that, okay, is it good enough to sell it in the market? Okay, sell it in the market. Okay, so can we think of any reason why there is a need for large scale production? Yes, the reason is going to be to make an n number of copies, okay, to make larger amount, to sell it and to have the money, okay, you need the money, right? So here, if any protein encoding gene is expressed in a heterologous host, okay, it is called as a recombinant protein, okay. So students, if we use a gene into a heterologous host, it will be called as a recombinant protein. The cells harboring cloned genes of interest may be grown on a small scale in a laboratory. Yes or no? I can grow it in a laboratory with having so many conical flasks, adding the medium, adding the DNA, allowing it to be in a shaker for some time and then growing it. I can do that. Okay. Okay, now the cells are being maybe used for extracting they already started thinking that let's discuss the job. Okay, extracting the desired protein and then purifying it by different separation technique. Now the cells can also be multiplied in a continuous culture system. Students, in continuous culture system, as I told you, there is not going to be a lag phase. Now see, continuously I am taking a class. Okay, there is no lag phase here. That means what? You are learning and learning and learning, and it is an exponential growth. Okay, it's an exponential growth. But imagine I took one class like 1 month back, or I took one class which is around 10 hours of lectures. Okay, after that 1 week I won't take any class. Now that is going to have a lag phase, because by the time you come to the next class, you're almost you have forgotten the way this teacher is teaching, or you have forgotten the syllabus that was taught. Okay, in the 10-hour lecture, I have done only half the chapter. Next 10 hours lecture, I am waiting to do the next chapter, and there was a 1 month gap. Automatically, a lag phase will come. That means what? All the resources that you had, you used it up. All the information that you had, you have studied. Now you are waiting for new information. So automatically you will get that lag. You'll be like, should I check another video? Should I go to another teacher? Automatically, same thing here. If it is a single, fed bioreactor where I am adding everything and mixing it and then waiting, then lag phase is pakka. But imagine it is continuous. In the middle I'm adding. Okay, I'm adding information. I'm telling this. It will never go to a lag phase. Is this clear? Okay. So here, continuous culture system wherein the used medium is drained out from one side and fresh medium will be continuously added. Okay, to maintain the cells in their physiologically. So small volumes of cultures cannot yield appreciable quantities of products. Yes or no? Okay, you are starting something small. You cannot expect tomorrow yourself to be Ambani. You have to know that today you start, tomorrow maybe 0 profit will be there, third day maybe 10 rupees profit will be there. So small scale. Okay. Now to produce in large quantities, the development of bioreactors. Okay, bioreactors where large volumes, 100 to 1000 liters of culture can be processed. Okay, was required. Thus, bioreactors can be thought of a vessel in which raw materials are biologically converted into specific products. Students, like how your mom mixes, no? With hand she'll be mixing the dosa batter, or with spoon she'll be mixing something. What she'll do? Imagine she would have added rice, dal, everything together, and she has grinded it. Okay, grinded it. She'll take a spoon. Well, that means what? She's mixing it. Right? She's mixing the rice, she's mixing the dal. Okay, then only tomorrow you'll eat dosa. Okay. So, bioreactors can be thought of a vessel in which raw materials are biologically converted into specific product, individual enzymes are going to be there, etc. Now, you can use a microbial plant, animal, or a human cell. A bioreactor provides the optimum condition for achieving the desired product by providing optimum growth condition like temperature, pH, substrate, salt, vitamins, oxygen. The most commonly used bioreactor are the stirring type, where continuous stirring is going to happen, students. Okay, now let's go further. Yeah, exactly. Okay, yeah, a stirred tank reactor is usually cylindrical. Okay, this is the image. Okay, this one cylindrical. Okay, so they are going to be cylindrical, or they'll have a curved base which facilitate mixing of the reactor. So what you have added? You have added the culture medium, you have added the bacteria, you have added everything, and you do the mixing, no? Like that, these people are going to do the continuous mixing at a larger scale, and we are going to get the product out of it. So they are going to be stirrer, they are going to be foam based, like a layer will be built up, no? Have you seen continuously if you mix something, slowly foam, bubbles will start forming. To remove that also they have techniques, okay? Now see, mixing the reactor content, the stirrer facilitates even mixing and oxygen availability. Students, this question has come. Okay, what was the function of stirrer? PYQ is there. So, question has come. The stirrer facilitates even mixing and oxygen. I think it was not a PYQ. It was there in one of the Vedantu mock test. I remember checking that paper. Okay, it was seen in the mock test. That is, what is the function of stirrer? The stirrer facilitates even mixing and oxygen availability throughout the bioreactor. So you shouldn't just write, I know when you think of stirrer, you'll be like, hey, stirring. Okay, stirring. Okay, yeah, but only writing stirring, you will get 0. Okay, you have to write that it also gives oxygen availability. Now alternatively, air can be bubbled through the reactor. If you look at the figure closely, you will be able to see that bioreactors have an agitatory system. Obviously they have to keep mixing, no? Obviously they'll have an agitatory system and an oxygen delivery system, a foam control system, a temperature control system, pH control system, sampling ports, and so many more. Okay, so if you look here, students, see, culture broth, sterile air, okay, flat bladed impellers, foam breaker. This is important because here there will be a layer of foam that will be formed. That foam will be removed with the help of foam breaker. Okay, then motor will be there, acid base to control the pH, it is there. Okay, steam for sterilization will be there, air is supplied from here. So everything is there in this. Okay, everything is there in this. Now look here, increase surface area for oxygen. How? This, like this, how we have microvilli, no, like this foldings will come and there more oxygen can come in because of continuous stirring. Okay, bubbles dramatically increases the oxygen transfer. Okay, so the bubbles will increase the oxygen transfer. Gas, so gas will enter through here. You can see gas will get built up. Okay, bubbles will be formed, then oxygen will come because they are going to make a, like a projection, like a furrow they are going to make. Okay, furrow they are going to make. Is this clear, all of you? Any doubts? Any doubts? Okay, so students, very simple. This is going to be single, okay, we can tell single step bioreactor or single, okay, bioreactor. What is going to happen? Everything you will give at one shot. Okay, now the graph that these people are going to have is going to be like this, students. The graph, oh my god, pathetic lines. Okay, this is one. How do I make copy? Oh no, copy. Okay, never mind. Okay, this is one graph. Okay, this is another graph. Okay, this you have to know so that you will be able to answer the difference. Okay, so this is a single step bioreactor where you added everything and then you just waited. So here what is going to happen? You're going to have a phase, you will have an exponential phase, then you will have a lag phase, then you will have a stationary phase, yes or no? Okay, so otherwise we can draw like this, a lag phase, and then a stationary phase. So students, this is the exponential phase where you saw the increase, where we got the product. Okay, where we got the product. This is the exponential phase. This is going to be a stationary phase or a lag phase. Okay, this is going to be the lag phase. This is in single. Why? Because everything has been used up. Okay, everything has been used up. Now at the same time, if you see it in a continuous, okay, continuously fed bioreactor where you saw, no, that they will remove the old stock and they will add the fresh medium. In the students, you will only be able to see an exponential. Okay, you will only be able to see an exponential growth because continuously medium is added. Okay, you will not see a lag phase. I have taught you everything in depth. I have not left anything. So all you have to do is just learn, download the notes and revise the notes. Okay, nothing I have missed, students. Extra information I have given so that you will be able to understand. Now students, upscaling we have done. Upscaling means what? I have increased my scale, right? Upscaling means now Sony and Rathis have upscale. That means they have increased it to 100 to 1000 liters. They have bought a bioliter that is going to, you know, culture, make the culture medium and the product from 100 to 1000 liters. Okay, before they were doing it in small conical flask. Now from here, next processes, I understood that okay, I did the upscaling, everything is done. Next I am going to do downstreaming. Okay, downstreaming. Downstreaming means I am going to check the product. Okay, I am going to verify or check the product if it is good to go to the market, because this too made, no, obviously I will check. Okay, see, after completion of biosynthetic stage, that is making of it, the product has to be subjected through a series of process before it is ready for marketing. So any product for that matter, okay, any product for that matter, you have to check. Okay, you have to check the quality of it before it goes to the market. Now, why do people get diseases, or why do people die because of food infection and other things? Because those people are not checking, right? They're not checking if the product is expired or not. From many restaurants, they have done, topmost restaurants, they have done, they have checked, okay, they have done rates, and they noticed that lot of expired products have been mixed in food. Why? Because they don't care about the marketing, okay. So same way, students, whenever we are selling something, especially such a complicated thing, like you are selling a desired recombinant DNA, recombinant protein, okay, we have to ultimately form a desired character. For that, if you want to sell, you have to make sure that everything is proper. Even if ligation is not proper, even if amplification is not proper, your entire process can go wrong, right? So here, what is going to happen? Okay, this is business, yeah. So now we will check their work, okay, because it is Sony and Rathis, we have trust issues, so we will check. So the process include separation and purification, okay. We have to give the most purest form, okay, purification, which are collectively referred to as downstream processing, okay, downstream processing. Now, the product has to be formulated with suitable preservatives. If you want to sell something, you have to add preservative to that, right? Preservative. Such formulation has to undergo through clinical trials, as in case of drugs, okay. Strict quality control testing will be done, okay. Strict quality control testing will be done for each product is also required. The downstream processing and the quality control testing vary from product to product. That's why I told you, like, now if someone else has made it, I will tell, okay, it's fine, okay, now this two has made it, so I will be like, sit now, I will check everything. So like that, okay, it matters different to different, joking, students. So understood downstream processing, you will not get a lot of questions on it, but understand what is going to come into downstream, okay. The question, I have seen a mock paper question again from Vedantu. Downstream processing, students, downstream processing, okay, downstream processing is actually where you do, what do you do here? Separation, purification, separation, purification, yes or no? We do separation, purification, that is downstream processing. After that, we add preservatives, okay, preservatives will be added, strict quality checking will be happening, okay, plus quality check. So they'll ask you, is separation and purification, you have to add as downstreaming process. Understood? Yeah. They'll put desired gene instead of using ligation, they'll put cement and they will stick and give. All of you one pieces. Telling that whichever character you want, you take. Okay? Yes? Done? Okay? Yes. Now ready for quiz? All of you ready for quiz? 2 minutes I'll sit. After that we'll be ready. Okay, 2 minutes all of you drink water and be ready for quiz. If you answer wrong, then it's there for you guys. Okay, so much I've taught in detail. Ouch. Yes, ready? All of you, full fire emojis in the chat. And did we reach the target, Madhumati? Did we reach the target? I don't think we reached the target. This lazy people hasn't made me reach the target. Such a poor target. Okay, questions? Okay, think and do all the questions. It's easy. You have learned it with me. Trust yourself, trust me, and let's do the. Okay, let's do this. Okay, this also is clear, no? Now, students, look here. Identify a character that is not desirable. Okay, not desirable in cloning vectors. Okay, what is not desirable in cloning vectors? Yes. Exactly, very disappointing. Okay, it's okay. Let's hope for the best. Okay, more students will come and join and see. Identify a character that is not desirable in cloning vector. An inactive promoter, an origin of replication site, selectable markers such as genes for antibiotic resistance, one or more unique restriction endonuclease sites. Okay, students, please think and tell, please think and tell. Okay, I can only see one correct answer. I can see so many wrong answers. What did I tell you? I told you that PBR322 has BAMH1, SAL1, PST1, PVU1. They are all 1. Okay, so here they are telling one or more unique. You should see this word. You shouldn't see the word one or more. You should see the word one or more unique. Right? One. Now, your PUC 18. It has around 13 sites. 13 sites are there. All the 13 sites are unique. They are more in number, but still they are 13, right? So look at the words. Words are going to play for you. Your exam is going to be easy. You have to understand the question. See how many of you did mistake. All of you went for answer 4 except Atuluru. Okay, I don't know how to pronounce your name. Yes. Okay, Sneha. Others I am not looking at because top I could see your answers. Okay. All of you now you are changing, but only he was the one who, you know, told the answer first. An inactive promoter. Why will you need an inactive promoter in a cloning site? You need an ORI. Yes, ma'am, I need an ORI. I need selectable marker like your tetracycline and ampicillin. I need 1 to more, you know, cloning site, that is like your PST, PVU, Eco R1. So the only thing that I don't require is an inactive promoter. Okay. Inactive promoter. Next question, it's very small. So, okay, I'll just read it for you guys. It's a question that I made you do. Okay, see, listen to me carefully. Okay, listen to me carefully. A cloning vector has 2 antibiotic resistant genes, tetracycline, okay, and ampicillin. A foreign DNA was inserted into the tetracycline gene. What I taught you, okay, desired gene, tetracycline gene. Non-recombinant would survive in a medium containing what? Okay, in a medium containing what? This is ampicillin, not tetracycline. Students, it's non-recombinant. Non-recombinant. Okay, remember this. Here it is tetracycline and not ampicillin. This is tetracycline and ampicillin. This is neither of them. Neither of them. Okay. But there's a lot. Yes. Can you answer? Yes. Answer? Yay, all ulta pulta answers you people will tell. I will hit you people. Think and tell the answer. I cannot hit you all together, no? Students, see, it is non-recombinant. That means desired gene is not inside this vector. Only vector is there. Only vector is there. So who is going to grow? Think properly. Students, listen. It is non-recombinant. Okay, non-recombinant means no desired gene. No desired gene. No desired gene has come in. That means what? Only vector is there. If only vector is there, it is a transformed, okay, it is a transformed non-recombinant one. These are the only ones that will grow both in tetracycline and ampicillin. Yes or no? Yes or no? This is why I told you people, listening is not enough. You have to understand every single word. Okay, first class, so it's okay. But look here, what is the question? It is transformed. Vector has come in, but they are non-recombinant. So see option number A, it grows both in ampicillin as well as tetracycline. So all your answers are wrong. All your answers are wrong. Understood? So this is how you decode it. Understood? So you should watch the lecture after this once again, after reading NCERT. Then only this chapter will be thorough. That is why students are not able to score that 40 marks from biotechnology. They are keeping the hand on the head and sitting. Okay? Those who join now, quickly mark your attendance, students. Come on, show some love. Now understood? Understood, yes or no? Nothing confusing, dear. Transformant. They are telling that vector is there. That vector has both antibiotic sites. That means what? It is transformed. Vector is inside. If they would have told that no vector is there and it is non-recombinant, you can blindly tell. Okay, you can blindly tell that they won't grow anywhere. Here they have told that the vector has antibiotic resistant gene and it has grown. Okay, only thing they have told is just non-recombinant. That means we inserted the foreign DNA, but it did not grow. It did not get transferred into the host. Understood? So, answer is going to be option number 3. Okay, answer is going to be option number 3. Okay, they told that insertion happened, but they are asking you what will happen to a non-recombinant one. Clear? Next one. Next one. Elution. Elution is what? Separating the restricted DNA fragments on agarose gel, staining the separated DNA fragments, cutting out the separated band of DNA from agarose gel and extracting them from the gel piece, constructing our DNA by joining the purified DNA fragments. Okay, think carefully and answer. You know the answer. You know the answer. You can trust yourself. Okay. Okay, good. I'm happy. I'm seeing some good answers. I told you, suction. Like suction means what? Removing that block. Other than Rohit and Sony, other people, what's the answer? Okay, Evening Star. Next. Why did Evening Star delete? It's correct? No. Yes, it is cutting out separated band of DNA from the agarose gel and then extracting the gel piece. Okay, it is option number 3. Okay, option number 3. Because see, separating the restricted DNA fragments, no. Okay, staining, no. Constructing, no. So it is cutting out or removing it out. Okay, ejecting it out like that. Now, when isolating the pure DNA from bacterial cell, the cell should not be treated with what? While isolating the DNA from the bacterial cell, we should not treat it with what among this? We should not treat it with what among this, okay? Not is the word. Not be treated with what, which among this? Very good, very good. Students, I want all of you to practice. If you have Tatva books, please take Tatva books and practice all the previous year questions. Okay, previous year questions. Yes, very good, very good. I can see all the answers coming. When isolating the pure DNA from a bacterial cell, the cell should not be treated with what? Okay, yes, very nice, very nice. D, yes, D is deoxyribonuclease. That means we shouldn't use a DNase. If you use a DNA, then the DNA will degrade. Right, DNA will get degraded, so we should not use a DNase. Clear? Next question. A host cell normally does not take up a foreign DNA until it has been made competent to do so. This is because of what? Simple. A host cell normally does not take up the foreign DNA. That's why not everyone can be the host, not everyone will be doing competency. Okay? A host cell normally does not take up the foreign DNA until it has been made competent to do so. This is because of what? Okay, this is because of what? Tell me. Because DNA is hydrophilic molecule, DNA is a very large molecule, there is no receptors for DNA on the cell membrane, DNA is an inert molecule. Okay, where will you guys use my name? Huh? You're telling all other teachers' name. Where will you use my name? Use my name also, no? There's no lag, dear. Yes, very good, very good. Ma'am, the answer is that DNA is a hydrophilic molecule and the bacteria is hydrophobic. So, they both will have issues telling that I like water, you don't like water, I should take bath, you don't take bath, blah, 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 they'll fight. So, to avoid that fight, we will use a divalent ion or we will do other vectorless methods to directly inject the DNA. Okay, perfect, good job. Next question. Taq polymerase, okay, Taq polymerase is used in polymerase chain reaction because of what? Okay, I think this is the last question. No, one more question is there. Okay, because of what? Taq polymerase is used. Okay, very good, Sony. Okay, I'll give you extra one mark. Taq polymerase is used in polymerase chain reaction because of what? Okay, yes, because of what? A, B, C, D, only 4 options they'll give you. A, B, C, D for till G they'll not give for my name to come. Okay, so it replicates DNA faster than other enzymes. It is the only enzyme that can replicate DNA in vitro. It does not require an RNA polymer to function. It is thermostable. Okay, easy questions. But there are, see, lot of mistakes you people are doing, but very small mistakes which can be rectified. Okay, which can be rectified. Yes, very good, very good. Good job. Answer is because they are thermostable. Many students will go and write this because it is the only enzyme. Who told it's the only enzyme to replicate DNA? We are doing DNA polymerase replication. We are doing replication. No, that time we are using DNA polymerase. Main reason is because heat cannot be withstand, right? DNA polymerase cannot withstand that heat. If it cannot withstand that heat, I cannot use it. Right, so I will use Taq polymerase. Last question, students, last question. The first type, this I had told you in the last class, the first type 2 restriction endonuclease discovered that could cut double-stranded DNA at specific site. Okay, specific site. The answer is there in the question only. But still, you should know it, okay? They will not ask you like this. They can just ask you the first type of restriction endonuclease that was discovered to cut double-stranded DNA also they can ask you. Okay, they can ask you even that. Yes. Very good, very good. It is hind 2. Okay, Hind. Okay. Hind 2 we'll tell. Okay, Hind 2 we usually tell. Okay. Hind 2. Okay. This is going to be the first type 2 restriction endonuclease. Okay, we are done and dusted with today's biotechnology chapter. Okay, for those students who were asking me about the syllabus, I will show you that as well. Just give me 1 minute. Students, let me also check if I showed you all the animations. Okay, I think this is the last animation. Because later I will feel bad that I did not show you this. Okay, this is the last animation. Okay, this was there. Okay, this one is there. See? This one, I showed you this, and I showed you this. Okay, done. Okay, done. How was today's class, guys? I want to see your comments because I found the class very fun. Did you all find it fun? Yes or no? So students, here it is. Okay, first link itself. Okay. So here if you put the phone number, students, even if you are taking the test, okay, I'll tell you one more thing which no one told you, maybe Shreyas would have told you. Even when you're taking the test, like you're like, ma'am, I'm not going to get scholarship, but I'm taking the test. If you're taking the test also, you will get All India rank performance and report, okay. Other than that, you will get prizes if you get the scholarship. You can also get scholarship in Vedantu offline as well as online batches, but you will also get free study materials, okay. For people who are taking the test, you will get free study materials. So at least for that, write the test. Okay, write the test and enter your phone number, then you submit the OTP. So dates are here, okay. Offline, it is going to be on 29th September, 6th, 13th, 20th and 27th October. Okay, it's very difficult to remember so many numbers. Online, it is from 10th to 20th. So almost 10 days this exam is going to happen online, okay. Scholarship up to 100%, okay. So identify your talent, understand your strength, highest personal attention and get inspired to write your success story. Clear? How is the class, all of you? Okay, I want to see the comments in the comment section. Okay, comment section. Got it? Homework question. Okay, homework question. If the DNA fragments undergo, okay, if the DNA fragments go through 7 cycles of your polymerase chain reaction, that is for amplification we use 7 cycles, then what will be the how much copies will we get? Okay, how much copies will we get? That's your question. So n is equal to 7, find the copies and let me know in the comments. Okay, so yes students, thank you so much for joining. See you all in the next class, and I will see you on Saturday with biotechnology applications. So now we have studied how to make them. Next class we are going to learn how to use them and where it is used, why it is used, and we will also understand Sony's and Rati's business exactly, because next is going to be application, and we will understand how this recombinant DNA technology has actually helped us in lot of things. Okay, be it a disease curing, be it about small fruits and vegetables, also we'll be understanding. Okay, so can I see all of your comments today in the chats? Yes, all of your comments. Yes ma'am, we can see it. Okay, so I loved the class. It was super fun. It was so nice teaching and making every small bit understandable. So let me know in the comments how the classes were. See you all. Thank you so much. Bye bye. Tata, bye bye students. Don't forget to comment. Okay.